On behalf of the Symposium Organizing Committee, I, Shifali Tyagi, and Iba Mateen extend a warm welcome to all of you to the 3-day symposium on Towards NTB, Achievements, Challenges, and Future Directions. We will start with Dr. Krishnan Mohan Atmankuri, Associate Professor, THSTI, who is also a part of the organizing committee, by providing a brief introduction about the symposium and its objectives.
Good afternoon. On behalf of the Executive Director, THSTI, and the Organizing Committee, I wholeheartedly welcome all of you to this wonderful, tightly packed symposium. The symposium has close to 400 plus participants, attending from 50 plus academic and research institutions and universities. In the next 3 days, a total of 120 plus poster presenters, 49 speakers, and 25 session chairs will intensely deliberate over 9 different sessions, the latest scientific details on different facets of tuberculosis, including the basic biology of the pathogen, its interaction with the host responses to infection and TB disease, TB vaccines, diagnostics, drugs under development, and TB clinical research. With close to 80 plus authorities, experts, and stalwarts of TB under this single roof, the organizing committee anticipates that the symposium will not just be an engaging and thought-provoking one, but also promote significant contributions, collaborations, and innovations towards the TB Mukt Bharat, as aspired by the Government of India. Understanding TB pathogenesis and the pathogen per se is central to the elimination of TB, and we have come a long way in that direction. TB has challenged humans and withstood our immune onslaught from time immemorial. While bioinformaticians predict that the pathogen evolved from its closest ancestor at least 15,000 years ago, the archaeological excavations from India and China date back TB association with humans to at least 2,500 to 3,000 years ago. From being first considered as a genetic defect that trickles down into specific families and dynasties, TB has later been classified as a type of cancer. Before, it was considered in the Middle Ages to be cured only upon the blessing touch by the royal families. TB has traveled a long way before its true nature was revealed by Robert Koch on March 24, 1882. The rest, as they say, is history. Until the discovery of the antibiotics, treatment for TB was limited to cod liver oil, vinegar massages, inhaling hemlock, turpentine, with warmth, rest, good food, and lots of sunlight, and the blessing of God. Between 1951 and 1966, 4 main drugs, namely isoniazid, rifampicin, ethambutol, and pyrazinamide, revolutionized the TB treatment and healing. However, to this day, even with this 4-drug cocktail, a patient requires at least 6 to 9 months of uninterrupted treatment. With the emergence of the MDR and XDR TB, though alternate TB drugs are becoming accessible, because of the extended treatment duration of up to even 24 months, despite implementation of the DOTS Plus programs, approximately 50% of the TB patients abruptly disrupt or stop their treatment altogether. Thus, unlike other infectious diseases, where the treatment duration is often 1 or 2 weeks, shortening the TB therapy remains one of the most sought after the global efforts, as this will be certainly a game changer in the treatment of TB as well as in the outcomes. Considering its central role in the NTB mission of India and the world, the organizing committee has aptly designed this 3-day symposium. Though globally, eliminating TB is very challenging, it is still achievable through a comprehensive, multi-pronged, patient-centric approach. The organizing committee trusts that the superior vaccines, timely but accurate and cheaper diagnosis, breaking transmission chains to prevent pathogen exposure, treating latent TB, targeting persisters, together with the shortened TB therapy, and minimal drug doses should move us closer to the TB Mukt Bharat. The THSTA envisions Nishchay se Nikshay, wherein Nishchay means with a strong will and Nikshay means TB-free world. This requires firm resolution and bringing together different stakeholders, sharing knowledge and resources, enhanced funding, and significant volume of the international and national collaborations. We sincerely hope this TB symposium will bring and provide such a platform and act as a springboard to steer all our scientific efforts to make India and the world TB-free. Lastly, as a token of pledge and for your sincere efforts in striving towards TB Mukt Bharat, we urge you all to sign on the banner, which is just placed at the entrance of the auditorium. We have just right at the doors. Again, on behalf of the organizing committee and THSTA fraternity, I thank you all and wish you your stay during the meeting enriching and comfortable, and hope this symposium will remain a catalyst for your innovations and research for a long time to come. Thank you very much.
Thank you, sir. We start this three-day event with the welcome address from THSTI's Executive Director, Professor Pramod Garg. Dr. Garg is a professor of gastroenterology and former associate dean of research at the All India Institute of Medical Sciences, New Delhi. He is an internationally renowned physician scientist with significant research contributions in the field of pancreatic diseases. He is a J.C. Bose Fellow and a Fellow of the Indian National Science Academy, Indian Academy of Sciences, and Royal College of Physicians. Sir, I request you to please give your welcome address for this event.
Good afternoon, ladies and gentlemen, honorable guests from India and abroad, our faculty, scientists, students, staff, and all the delegates who have come for this three-day conference on tuberculosis. It's my real pleasure to welcome you all to this institute and to this wonderful meeting. I'm very happy to see the hall full right in the beginning, and that shows the amount of effort which has gone into organizing this event. When we discussed about tuberculosis, during a conference symposium on tuberculosis, all the faculty and scientists who are working on tuberculosis at this institute were very happy and forthcoming. And it so happened that we are organizing it on March 24. Actually, today is a kind of a pre-function day. So it was very apt to organize during the TB Day in India. As a physician, we have always been struggling with tuberculosis, both for diagnosis as well as treatment. Although I must say that if it is diagnosed well in time and you give appropriate therapy for optimum duration, more than 95% of patients can be cured. And research from India, particularly from Madras trials earlier on, Dr. Swaminathan is here, that many studies have been conducted which have shown that recurrence rate is actually very low. The problem is that if the treatment is the disease is resistant or the treatment is not continued, then there are many problems. And in the clinic when we sit, we always keep tuberculosis as one of the differential diagnoses. They say that an uncommon presentation of a common disease is more common than a common presentation of an uncommon disease. And tuberculosis being so common, it is always in our differential diagnosis. In fact, whenever you have a CPC, clinical pathological conference, and there is a very esoteric, very uncommon case being discussed, the discussion will always keep tuberculosis as one of the differential in any discipline. So that is the way tuberculosis presents, and that is how common it is. So I think it is time that we renew our efforts in eliminating tuberculosis from our country. The SDG goals say it should be by 2030. Honorable Prime Minister has given a call to end it even earlier. And I think if we have more and more efforts from different directions, there is no doubt that we should be able to at least control and stop new cases emerging. At THSTI, I'm very happy to tell you that there are a lot of good science being done, both in the realm of basic biology of tuberculosis bacilli and also drug discovery. And Ramandeep, Nishit, Amit, and Krish, and many other scientists are working tirelessly to come up with new therapeutic molecules. And I think today we have 4 or 5 such molecules which are ready to go, in a sense that they are just waiting for some more results and they can be, phase 1 ready. And that is the reason why I think a conference like this, where we have participants from academia, from industry, from other stakeholders, is so very important, because connection between academic and industry is essential if we have to take one of these molecules forward. And that is where our country has been lagging behind in all the areas of drug discovery. But COVID has actually shown us that we can do it ourselves, vaccines. And I'm happy to tell you that at THST also we have a new vaccine coming up, which in partnership with industry, which is going to go for phase 1 trial soon. So in drug discovery also, I find a lot of strength, in terms of biology, in terms of science, that a lot of strength in India. And if it is at THSTI, I'm sure there are many other institutions in the country which also have similar molecules, similar therapeutic, targets. So having people on board from different industry and academia and other, even ministry will be very, very helpful. And Dr. Swaminathan was telling me that she is making some more efforts to have a larger dedicated, pool of people who can actually contribute to more successes in this area. The program I found is actually power-packed. It is running up to some 8 p.m. or something. So I'm not sure if it will obviously spill over to your dinner time. So instead of 3 days, it could have been 5 days. I think they wanted to save some money on hotel and other things. So they compressed it in 3 days. And they are very ambitious, but it's fine. So thank you for making all the efforts and getting all these star speakers for this very important conference. So my best wishes to all the delegates for happy learning. And I'm sure this effort will go a long way in solving the tuberculosis problem in India and globally. Thank you very much.
Thank you, sir, for your warm welcoming words. Sir, I request you to please be seated on the dais. I now warmly welcome Dr. Soumya Swaminathan, former Chief Scientist at the World Health Organization and currently the Chairperson of MS Swaminathan Research Foundation, Chennai, to deliver the first keynote lecture. Dr. Swaminathan was also a former Secretary to the Government of India for the Department of Health Research and Director General of the Indian Council of Medical Research. In addition to being a pediatrician, she is globally recognized for her research on tuberculosis and HIV. She was the former director of the National Institute for Research in Tuberculosis, Chennai. She brings with her more than 30 years of experience in clinical care and research and has worked throughout her career to translate research into impactful programs. Following the keynote address, there will be a 5 minutes Q&A. In the interest of time, I request the audience to keep your questions brief so that there's an opportunity for others. Ma'am, I request you to please share your valuable insights with us.
Good afternoon, and thank you very much for the warm welcome and nice introduction. And congratulations to the team, Raman, Amit, Krish, and others for having put together this really excellent program. And you can see the interest also. And it's wonderful to see so many young people in the audience, because I think one of the things we need is more young people in TB research. It is a difficult area, and it's one of those areas where you can easily give up because progress seems to be slow. But unless we have more energy, more creativity, more ideas in this field, we're not going to make progress. So really special greetings to all of the young people here. So this is this year's World TB Day theme. Yes, we can end TB, because I think WHO wanted to have an optimistic and a positive message out there, because very often when you look at TB statistics, you can get extremely depressed. Of course, what happened with COVID is that we saw a lot of backsliding in TB as we did in other disease areas, because all of the resources and attention was focused on COVID in the last 3 years. So for the first time, actually in 2020, we saw an increase. We saw a decrease in case notifications. Normally, one expects about an estimated incidence of 10 million cases. We were almost at 7 million notifications. Over the last decade or so, things had improved, a large part due to India, actually, where a lot of private sector notification had increased. But then again, there was a backsliding, and you can see here in 2020, fewer people accessed TB care. So there were only 5.8 million reported cases. So almost 1 million plus cases never got diagnosed. And so 4 million cases worldwide, and we had 1.5 million people dying. And as you know, TB is a top infectious disease killer. And we heard from Krishna a very nice historical overview. Now, if you look at 2021, it got worse. So 1.6 million people died of TB. And here were some of the NTB strategy targets actually that were set, which was supposed to be achieved in 2025. And you can see that we were supposed to reduce incidence by 50% between 2015 and 2025. Well, there's been hardly any reduction, 10% last year, but I think you'll see that it goes down even more because of COVID. And similarly, on deaths, we were supposed to have made significant reduction, 75%, but you can see there is negligible change. In fact, as I told you, deaths increased in 2021. And the third goal was on the zero people with facing TB catastrophic costs. And as you know, again, many of those people live in India, and they get pushed into poverty when they get TB because of all the expenses on getting themselves diagnosed and treated. So it's clear that what's needed is more funding, more research, more innovation, and of course also translation of those things into the program. And so these are the reports produced every year by WHO, but what we saw was that there's supposed to be $13 billion spent annually on providing access to TB diagnostics treatment for people, and only 5.4 billion was spent in 2021, not even half. And normally it's around 6 billion, so it went down again because of COVID. And on TB research, there was a very conservative target of 2 billion. And if you can think that in COVID, at least 100 billion was spent globally just on developing vaccines. You know, within a year and a half, 2 years, the world spent $100 billion. And it obviously showed results. You got many, many dozens of vaccines. But I think that we have never touched even half of that very conservative investment in TB research globally. And this is data that, gets collected from research funders around the world. Below is the TB preventive treatment targets. And again, except for people living with HIV, where thanks to South Africa, mainly these targets were met. If you look at preventive therapy for children under 5 or household contacts over the age of 5, they were very, very far from meeting these targets. Again, on treatment also, there was a UN high-level meeting in 2018. There was a lot of global commitment by political leaders, and there were targets set that we need to treat 40 million people, but only 26 million were treated up to last year. So that's 66%. And again, every time you come to children, everything is always worse in terms of meeting the targets. So if you look at the risk factors for new TB cases, this is important because while we try to diagnose and treat as many cases as possible, ultimately, you want to understand how to prevent. Undernutrition is the single biggest risk factor. And in India, it accounts for almost 50% of our new cases. Just one factor, and that is undernutrition. And so a BMI, a low BMI is the biggest risk factor. And this has been borne out by the National TB Prevalence Survey that was just completed in India. It was started before the pandemic, so there were a bit of delay, but it was completed in 21. The results were, I think, not yet published, but certainly have been presented. And again, the same risk factor showed up in the India prevalence survey, but a low BMI was over and above, just like you see on this graph. And then you have HIV infection. Clearly for sub-Saharan Africa, that is the major risk factor. Alcohol, smoking, diabetes. There's been talk about air pollution, but no conclusive evidence on that. And so what happened with COVID is that TB deaths increased, fewer people were diagnosed and treated, and there were fewer resources, both for essential services and for TB R&D. Now, this is some modeling that was done by Nim Ariman Pati and colleagues from the Imperial College in partnership with the ICMR. And this is looking, modeling to see like what interventions would make TB incidence go down. So you see that's the baseline. And then if you do improve diagnosis, then you can reduce the incidence. If you do accelerated case finding, then you can make a significant impact with preventive therapy. And then if you do mass vaccination with an effective vaccine, then of course, that's the best way of achieving the target. And by 2030, then all of this, you can achieve the NTB targets. But this is what COVID did, big spike in cases and deaths. And therefore, we would need to accelerate. And even with acceleration, it looks like even the 2030 targets are a bit difficult. And these graphs actually depend on finding these new tools. If you had a molecular diagnostic or a rapid point of care test, that would make a big difference. Similarly, an effective vaccine would. This is just a study which looked at co-infection with TB and COVID-19, and it was found that there was a higher mortality, and this was associated when there was a co-infection with older age, male gender, and ventilation. So TB patients are at high risk for COVID. During the pandemic, we saw a huge expansion of the use of digital tools across the board. And this was true for TB also. Lots of digital solutions, apps, chatbots, risk assessments, referrals, contact tracing. So people got quite imaginative in the way that these digital tools were being used. And many of them, I think, are very useful for the future as well. We, of course, are seeing a lot of progress in the use of artificial intelligence. And in the case of TB, one application is to do automated X-ray reading. And today, with the ultra-portable digital X-rays, which you can literally hand carry, it's just slightly bigger than a normal camera. And linked with an AI-based reading of the X-rays, you can see that you could go into villages and screen, or into high-risk urban settings like overcrowded slums and so on, which gives you the opportunity for high-throughput screening. Similarly, lots of work going on now on using AI and digital stethoscopes that could be used just by analyzing cough patterns. This was again tried for COVID as well. So if you could, from the cough pattern, distinguish between different types of diseases, then that's another useful application. So some of these could be used for initial screening because they could be done easily, cheaply, quickly, and they will never be confirmatory, of course. And then you have to go and do a confirmatory test, but this may give an opportunity for us to actually find all those missing cases. So when we start thinking now about diagnostics, therapeutics, and vaccines, I'll just say a little bit about each of them. I know you're going to hear a lot in this conference about basic science, so I'm not going there, but clearly basic science is extremely important for us to make fundamental progress in understanding TB. And the problem is we still don't understand the entire life cycle pathogenesis of TB and how it lives with the host. In many of us, TB bacterium lives very comfortably for our whole life. whether it's dormant or in some form. And there are certain life situations where it gets reactivated. And in others, they are very prone to quickly progress to TB once they see an infection. So host immunity is something that, and the whole concept of the granuloma and how Mycobacterium survives intracellularly, that is really the ultimate holy grail. The other, of course, related to that is the immune system. And again, I think if you look at, compare with COVID and the huge amount of work done in immunology in the last 2 years and how much we understand about, I don't think it's unprecedented actually, the amount of progress that's been made, especially both in virology as well as in immunology and understanding CD4, CD8 T cell response dynamics. So those things are going to help us. And I think some of those advances, technologies and methods that were developed should and could be applied to TB. So in the case of diagnosis, for example, we need to now start thinking, unfortunately, 4 out of 5 approximately patients in India still have only access to microscopy and not to a molecular test. And this is a bit shocking considering the expansion of molecular platforms across India and the availability today. So it's just that they've not been deployed for TB. In fact, many of the TB gene expert machines were deployed for COVID, which is why case finding went down. So every suspected TB case or somebody with symptoms must have access to a molecular test, because that is the test which is considered the gold standard today. And of course, there are different platforms and it has to go through regulatory approval. The good thing is there are many companies now in India that are working on alternatives to the gene expert, which has been more or less sort of a monopoly in the global market now for many, many years. And that is never a good thing. So hopefully we can see the price coming down without, without quality going down, of course. So that's the first paradigm shift, is that we should not be satisfied with a smear microscopy result, because we know you're going to miss 50% of even infectious cases. The second is that we need to move from centralized lab-based nucleic acid amplification tests, the big COBAS type machines, to decentralized POC NATs, and that's coming. Many companies working on it. And this would also go well with moving into multi-disease testing, where you have these diagnostic platforms at a primary health center level, which do not only TB, but also do dengue, do scrub typhus, do, whatever else are the common infectious disease. And those are the kind of platform technologies now that one would want to see in the health system. Of course, we need to move from sputum to non-sputum samples, because sputum is a very difficult sample to produce and to handle. And that's why we need oral swabs, urine or blood. And again, the testing needs to move away from what is passive when a patient has to go to a healthcare setting to closer to communities. And I'll tell you the reason why. The data from the prevalence survey tells us, why we have to do that. Then we also need, of course, a focus on accuracy is important, so specificity. But as I said, there are many screening tools now where you can cover large populations and move towards lower cost, high volume manufactured LMIC. This paper that I wrote with Madhu and Puneet is in press in Nature Microbiology. So maybe I'll just take a minute. I don't have slides from the TB prevalence survey, but I can tell you that in terms of findings, it showed that the prevalence survey was set up to get a national TB prevalence, point prevalence for the country as a whole, but also the sample size was such that it was supposed to be able to estimate the prevalence of at least 20 of the states, the largest states. So sample size pretty big, 3.5 lakh. That's 350,000 people, probably one of the biggest surveys done globally, or the biggest anyway. It was interrupted by COVID, but finally it was completed. And they looked at people who have culture positive or microbiologically confirmed TB. Of course, there were symptom screened, then X-ray, and then sputum. So it was only pulmonary TB that was looked at. And the finding was that the prevalence was about 310 or 314 per 100,000 people. I remember starting my career in TB with a very similar prevalence estimate, maybe a little bit higher than this, but certainly this is only pulmonary. By the time you add extra pulmonary and all that, you get close to 4 per 100,000, which is what I was told when I started my career. The TB prevalence in India is 4 per 1,000. That's 400 per 100,000. So in 30, 35 years, we have not made the progress that we should have. Second, and so the question is why? Second is that the prevalence survey showed that of 100 people who were found to be culture positive, 65 of them did not know that they had TB, had not taken either sought care or taken treatment or been diagnosed. So 65 out of every 100 people are going around in the community with TB without knowing it for whatever reason. Either the symptoms are still, not good enough, or they've just gone to their local pharmacy and taken some cough syrup and drugs. We know there's a lot of that goes on, self-prescribing, pharmacists prescribing, and there's also the possibility that they went but didn't get diagnosed. So the combination of these three, but the majority of those 65% were people who never sought care. They didn't know that. So how do you deal with a disease when 65%, there's such a huge community pool of cases, right? This is why I think the mass screening with X-rays and other initiatives for screening are going to be important. And you can, when they looked at risk factors, it was men over the age of 65, and then undernutrition, followed by diabetes, smoking, etc. That were the risk factors. But if you look at the graphs, you clearly see a sudden spike in men over the age of 65. So for some reason, that age group is very, very, and you know that men are 3 to 4 times more common than pulmonary TB is more common than in women. So the role of X-rays became very clear from that prevalence survey. It also showed that only 1 in 4 or 1 in 5 people had access to a molecular diagnostic. And it showed that these were the risk factors. Now, of course, there were statewide differences with, states like Kerala and all very low. But I think the sample size was not big enough to confidently say, which states had what prevalence. And hopefully those data will be published soon so that we can do more analysis and come up with some policy recommendations. Now, in terms of drugs and drug regimens, there has been progress in the last couple of decades. It's slow progress. There are molecules in phase 1. There are some in phase 2. This slide actually gives you both drugs and regimens. So you can see some of them are the names of trials that are trying to either shorten treatment or trying to use combination of newer drugs. So there are many, quite innovative designs today, including designs to see if you can treat some people with a very short regimen. So if you have, minimal TB, can you be treated with a 1 month or a 2 month regimen? And if you have cavitary TB, then you may need even more than 6 months to prevent recurrence. So those are the kind of optimized regimens. And of course, the ultimate goal is to have a universal TB regimen, because as you know, today we have different regimens for drug sensitive and drug resistant TB. A universal regimen would mean that you have all new molecules for which Mycobacterium TB doesn't have resistance so far, and then combine them so that you don't have to bother with drug resistance testing. You can treat everybody. Similarly for TB, latent TB infection, it's called by many different terms, is still an enigma. The fact that we used to talk about a quarter of the world, a third of the world having latent infection, because if you go around doing a skin test or an IGRA test, then obviously many of us in this room will be positive, especially the older people. But does that mean that we are at risk of TB? Not necessarily. In fact, no. If I've had latent TB for the last 25, 30 years, I'm not at higher risk today of TB. Whereas if you've just recently acquired an infection, then you are at very high risk in the first 1 to 2 years after infection. So those type of differentiations now need to be made, and there needs to be more understanding. And I know there are people here who have been working on biomarkers or risk indicators. How can you tell between two people who are IGRA positive, who's more likely to progress to TB and in what time frame, and how do you target them? And I know we have Andreas from South Africa, and South Africa has been also doing a lot of work in this. I have to mention also Cape Town, the studies that Robin Wood is doing on looking at TB transmission. This applies to what I was saying earlier about people in the community walking around with TB. So he is looking in the Cape Town settlements at putting in machines right there within that sort of slum. I don't want to call it a slum, but it's a low income place where he's capturing bioaerosols and finding that, again, 60, 70, 80% of people walking around in that area, you can actually grow MTB from the bioaerosol. So I think this is something new. We need to replicate that. We need to understand what does it mean. So here we have lots of trials for shorter preventive therapy regimens. And again, if you look at the vaccine pipeline, well, it's not changing a huge amount, but we do have candidate vaccines. And we have Mark Feinberg here from IAVI, who is progressing the MTB vac candidate, hopefully with Indian collaboration. And we also have the Serum Institute and the VPM, which came from Stefan Kaufmann. It's a recombinant BCG. And then you have the M72, which has finished a phase 2B, but not progressed beyond. That came from GSK and is now with Gates MRI. And then there's a BCG revaccination trial also that Gates MRI will be doing, and a couple of other candidates perhaps earlier, and this may be incomplete. But there are obviously candidates that are targeting prevention of infection, prevention of disease, and prevention of recurrence. So one thing that would be useful for us to think about also is how do we share genomic sequence data for TB? We know that we have different lineages, we have different drug resistance profiles. This, we saw in COVID that the GISAID data sharing platform allowed us to share literally millions and millions of sequences, and we were able to track variants. Now, of course, MTB doesn't, change and mutate like a virus would, but it would be still very helpful to have a global data sharing platform. I know that DBT is setting up or has set up for India a sequencing platform to pool and share data. We need to now think globally. Can India take the lead and really establish? And GISAID would be very happy, I know, to collaborate with us if we wanted to do that. So we know that there has been progress in research. It's not that there has been no progress. A lot of new diagnostics have been recommended. We know treatment now can be shortened to 4 months for drug sensitive and to 6 months all oral for drug resistant TB. We now have short preventive regimens like 1 month or weekly for 12 doses. And we've had some vaccine candidates progressing. There's been a call now mainly from civil society and patient group advocates that we should go for this 1-4-6 initiative. That is 1 month preventive, 4 months for drug sensitive, 6 months for MDR. And they're calling on all countries to implement this because this is the patient friendly approach. And again, they have quoted what Paul Farmer said, calling for staff, space, systems, and support as a human rights imperative. A couple of closing remarks now on the lessons from the pandemic. And I was very much involved in coordinating some of these efforts at the WHO. We know that new platforms, which we had not heard of before, were deployed. And so we should be looking at those viral vectors, mRNA, maybe DNA as well. We did have to mobilize and deploy large-scale funding. As I mentioned, $100 billion approximately was spent, mainly by countries and some by private sector. There was a lot of harmonization of efforts between industry and research, institutes with WHO taking the lead in setting some of the benchmarks, developing the target product profiles, and aligning regulators. Very important. There was regulatory input into trial designs from the beginning. The developer knew these were the benchmarks to meet if they wanted regulatory approvals. deployment of efficient trial designs, including standard protocols, multi-site, multi-country studies, adaptive trial designs. There was accelerated regulatory review, record time, for licensure. The vaccine manufacturing capacity just, expanded scale dramatically. Of course, equity, access, available issues that we saw were not dealt with very well. And we need to keep that in mind. Any new product we develop for TB is a disease of poor countries. We have to keep access, equity, affordability in mind right from the beginning, and community engagement. We saw the anti-vax movements for COVID vaccines. Well, this is here to stay. We are going to see these kind of anti-science activities happening, and we need to keep in mind how to deal with that. There are now a lot of interest in mRNA-based TB vaccine candidates, some companies, including in India, working on this. The advantages, of course, are that you don't need a biological, manufacturing. You need basically, it's chemistry rather than biology. So it makes, to that extent, things easier. It's fast. You have the genetic sequence. You can quickly develop. If you have a platform, you can put it on the platform. Of course, again, for TB, it's a question of which epitopes, which antigens do you select. You can reduce the cost to generate clinical trial material. But the big but here is, without correlative protection, we can't say, like for COVID, very quickly, we knew that neutralizing antibodies could be used, at least as a broad correlative protection. We don't have that for TB, so we have to do the efficacy studies in humans. And there are still some unknowns for the mRNA platform that need to be looked at and evaluated. Now, I'm not going to go through this very complex slide, but I just want to say that the social determinants, and I talked about undernutrition, but there are poor working conditions, low socioeconomic status, food insecurity, stigma, discrimination. All of these things will contribute. And so that's why the SDG framework is useful, because it tries to address also upstream the determinants of TB. So I think that our response to TB cannot be the same as a response to any other disease. It's the #1 infectious disease killer. It affects people mainly in the developing world, in the poorer countries. And within those countries, it's the poor who are disproportionately suffering. And therefore, our response and our approach to research has to be different. And so a few of us wrote this paper in Nature last year, where we said that a model for R&D, which is addressing big public health challenges, should be different. It should prioritize public health needs. So for India, for example, TB would be very high up. Perhaps there would be things like dengue as well. Malaria is still a problem, of course, NCD. So we need to do that prioritization. The R&D has to be, of course, scientifically strong, and it has to be ethical. We need to incentivize and facilitate rapid open sharing of processes, inputs, outputs, like we did for COVID. This is why we were successful, because people were willing to share. Nobody was sitting and waiting to publish their findings. At the WHO, on a weekly basis, we saw researchers sharing the latest data without really, and of course the journals also cooperated. They said that you can put your paper into a preprint and we will still publish it. So that is very important. Timely access to health technologies. So we again set up this technology transfer program when I was in WHO, which enables the transfer of technologies like mRNA to countries that do not have access. And as you know, the big pharma companies refused to share their IP or their know-how. And so we said we would do this anyway in a very open and democratic way. So it's something, I suppose, like open science, where you use a new technology to develop the products that are most needed, and you share it with people who are in a similar situation. So there is now a network of 16 countries, India is included in that, which I think are meeting in Cape Town in a couple of weeks to discuss how this mRNA technology can be used. And it's the help of Pfizer or Moderna, but we still were able to recreate the Moderna vaccine in South Africa, just in an academic lab, just from what's publicly available. So it's possible to do. We need to make sure that R&D meets the needs of subpopulations, children, pregnant women always, at the back of the queue, and then recognizing contributions and sharing benefits equitably. There's a lot of discussion now on access and benefit sharing that's happening. But as I said, again, the bottom line is that for a disease like TB, affordability, availability, suitability are very important, and they need to be thought about right from the beginning. This can only happen if governments, public, private sector, civil society come together and do this in a sort of mission mode. So let's hope that this conference galvanizes some of that thinking and brings people together. Thank you very much. Thanks. You wanted to take questions? Yes.
Thank you very much, ma'am, for your far-reaching insights on how to stop TB and make India TB-free. The floor is now open for any questions.
Excellent talk, Dr. Swaminathan. I have only one question. What about the incidence of reactivation in patients with COVID-19 infection? You mentioned about mortality, that mortality
That's
was
an
higher.
excellent question, I think. I don't have an answer to that. There may be somebody in the audience who's done that kind of follow-up, but it's a very important question, yes. What does post-COVID do, and does it exacerbate TB reactivation?
And Dr. Somil, thank you for that great lecture. So my question is pertaining to the modeling that you showed. What was really dramatic in that just the addition of vaccines has the largest effect. So these vaccines are so in the modeling, is it taken into account that these are childhood vaccines replacing BCG, or is it something that will be given after BCG? And either way, what should be the efficacy to meet that target?
It's a good question. I think we'll have to go back to that paper and that model to see exactly what attributes they used. But I think they used a vaccine that ideally you would want a vaccine that blocks infection, right? But even a vaccine that prevents progression of disease from latent to active, if given in adolescence, let's say, the beginning of adolescence period, I think that's the kind of vaccine that was used. With or without a BCG at birth, because right now everybody gets a BCG at birth in most countries that are TB endemic. So any vaccine that's introduced would probably be on top of that, for now anyway. But I think there are different approaches: neonatal vaccines, adolescent or young adult vaccines. So the thing is that vaccine would really need to reduce progression of disease to be that effective. And we know for any infectious disease that we hope to eliminate without a vaccine, it's going to be extremely challenging.
But any idea what percentage efficacy we're looking at to get such a dramatic effect?
No, we'll have to look into the details, but it would have to be high, at least above 70%.
Thank you.
The WHO did some modeling, so you might want to look at that paper as well that came out in December. And I think they said that even if a vaccine with 50% efficacy would make sense in a high burden country like India.
Hey, Dr. Swaminathan, very nice overview on the TB. So my question is, you talked about new vaccines. Since we don't know about correlates of protection in tuberculosis, so you'll have to go for efficacy studies. What I've been following, the TB vaccine programs, that we go for efficacy studies, but there's not really very serious effort on understanding the correlates of protection. So, as a clinician, your perspective is very important. Why a very large scale effort has not been taken yet, and what it required to really understand the correlates of protection in tuberculosis? Because I think without that, no matter how many vaccines we keep trying candidates, I think we'll be far away from reaching the success in that.
It's a very good question. I don't know, Mark, if you want to comment on that, but clearly there's an effort needed. And I think the reason why it's not happened is just, I think, lack of coordination. And I know Gates wants to do that, but IRB, certainly this is one of the goals they have. And the Gates MRI is trying to put together a protocol where everybody can test for surrogates. But Mark, yeah, please.
Yeah, no, I mean, as you know, to identify correlative protection, you need an efficacious vaccine signal, or at least a partially efficacious vaccine signal. And there is work going on in the follow-up to the M72 vaccine study that you mentioned that is a partnership between the Gates MRI and IAVI and a number of other partners. That's really kind of open source, so people who have ideas that they want to explore are able to apply to get access to samples to do correlates analysis, and that work is going on. But there are still questions about whether the range of samples or the assays that are available are sufficient right now to really truly identify correlates from what was a relatively small, proof of concept study.
Thank you, Dr. Soumya. Just one quick question on the TB discovery and development pipeline. To me, it sounds very positive and lots of new compounds. I would like to have your comments on the opportunities in host-directed therapies versus the small molecule drugs, if you have a thought.
Yeah, thank you. I didn't talk about host-directed therapies. Clearly, there was quite a lot of interest. There is still. I know at the NIRT in Chennai, for example, study was done using metformin. It was a phase 2A kind of a study that's didn't show enough, I think, benefit for it to progress. There was another phase 1 study looking at verapamil, because verapamil is a drug that that acts on the tolerance to rifampicin and some other antimycobacterial drugs. And so tolerance is a phenomenon also that leads to delayed response to treatment. Now, the issue with verapamil is that there's a drug interaction with rifampicin. And so verapamil blood levels are very low if given with rifampicin. So you have to give a very high dose to overcome. Or we have to manufacture like norverapamil or one of the other forms of verapamil. But those are the trials that we need, actually. There are many hypotheses now on host immune modulation that could help. There are new pathways that have been described. So if we could have a platform for clinical trial that just looks at host-mediated immune response, host-mediated interventions to improve TB treatment outcomes, it would be good. But, again, the sample sizes and all those things are complex, because with standard chemotherapy, you get very high cure rates. And so you would need to either have a very large sample size, or you have to pick MDR-TB or somewhere where you have poor outcomes in order to significantly show a difference. That often becomes a challenge with these host-mediated. I think there is scope for trials.
Yeah, hi. Just last question.
I think last question.
So my question is more about the connecting the host metabolism, especially now you have also indicated that nutritional associated phenotype correlate with the high disease prevalence and the mortality. So on that note, what is the prescribed regimen for managing the host metabolism, but kind of leading to the improved nutrition of the patient, So is there any standard that we know that when we should follow for enhanced efficacy?
Yes. So if I'm understanding your question correctly, it's about managing undernutrition and associated host metabolism. I mentioned undernutrition as a big risk factor for TB, but what I didn't say is that it's also a big risk factor for poor outcomes, high TB mortality, lapse, and therefore managing TB treatment is important. The TB prevalence survey did show that mortality rates are higher than what was expected or what is reported, because many patients die at home and the mortality is never reported. There are also deaths which occur right after the completion of TB treatment, which are not counted. But some states have actually moved to doing like assessment of all TB cases to identify those who are at high risk, severe TB, and simple things like BMI, respiratory rate, oxygen saturation. So 4 or 5 such parameters where you can identify the people you can classify as severe TB, maybe hospitalize them for a couple of weeks, give good nutrition, give oxygen, whatever they need, supportive treatment, and then send them home. There was also a trial done in Jharkhand called the Rations trial, which randomized families to receive nutrition support for just the patient or patient and family. That trial has not yet been published, but showed a good response to those who got nutrition. And furthermore, the contacts, the children who got additional nutrition had less of conversion, of TB infection from negative to positive. So I think the role of nutrition to prevent TB reactivation, but also once somebody has TB, is extremely important. There isn't enough attention paid, and there's probably some unnecessary mortality now that can be reduced.
With this, we'd like to thank Dr. Swaminathan for an excellent talk, telling us where the global efforts and funding have fallen short of expectations, but also giving us hope with the new vaccines and new therapeutic molecules on the horizon, which might actually, let us achieve our target. Thank you very much for this talk. Over to you.
Thank you, everyone. I now request Dr. Pramod Garg to felicitate Dr. Soumya Swaminathan. Thank you very much, sir. Dear ma'am and sir, I request you to be seated on the dais to chair the session 1. Today, we have 2 very exciting sessions, session 1 and 2 on TB clinical research that are interspersed with keynote presentations. Clinical research is one of the most important links for any bench discovery to be tested and evaluated in a population before implementing the same at bedside. It also serves as a primary link for unraveling the patient's clinical problems to the bench researchers for continued discoveries. The first session on TB clinical research will be chaired by Dr. Soumya Swaminathan, Dr. Pramod Garg, and Dr. Sarman Singh. I request Dr. Sarman Singh to also be seated on the dais. Dr. Singh is currently a professor and mentor of the Medical Science and Engineering Center, ISER, Bhopal, and former director and CEO of AIIMS, Bhopal. He is a fellow of 7 national and international academies, including Royal College of Pathologists and Royal Society of Chemistry. He has been awarded by several national and international scientific and academic bodies for his outstanding contribution to tuberculosis research, including the Dr. B.C. Roy Award by the Honorable President of India. We have 3 talks in session 1, each for 20 minutes. Following the 3 talks, there will be a brief Q&A session with all the 3 speakers. I request the audience to hold their questions until then. Also, I urge the speakers to stick to their time. The buzzer will ring at 18 minutes. I request the speakers to be seated on the dais after their talks for the Q&A session. I now request the session chairs to kindly lead the session 1.
Thank you. So on behalf of the co-chairs, I think we'll get right into the session because everybody is eager to hear from our speakers. Our first speaker is Dr. Camila Rodriguez from the Hinduja Hospital. She's a consultant, microbiologist, and chair of the Infection Control Committee. I think everybody knows her and her contributions in the field of TB, especially in the area of diagnostics, drug resistance. She's had over 350 publications. She's extremely well known. And she's a good friend with Camila. Over to you.
Thank you, Soumya, and thank you, organizers, for inviting me to this wonderful 3-day symposium on the commemoration of World TB Day. I think the poor accuracy of smear microscopy and the weeks to months long time for culture has really been the biggest impediment to this long wait for the diagnosis of TB. It really has been the Achilles heel. And there are decades when nothing happens, and weeks when decades happen. And I think that is actually applied to the TB diagnostic pipeline. It's suddenly got richer and better. And this is the FIND pipeline study, which is just showing that over 2019 onwards, every 3 months, there's something really new, exciting on the horizon. And yet, testing is the weakest link in TB care. Just 1 in 3 patients are confirmed bacteriologically, and 1 in 5 are molecularly confirmed by the WHO-approved tests. And even with drug-resistant TB, it's tragic that just 1 in 3 patients are put on treatment, despite WHO endorsing a huge number of tests. So it really was time to usher in a new era, and molecular diagnostics with genomic testing really is the order of the day, as Soumya clearly said. But I think we need to understand the limitations of the gold standard that we sort of hold so close to our heart. Apart from being slow, phenotypic DST requires positive cultures. But to me, the greatest fault, if I can use that word, is that we test at a single binary critical concentration at inappropriately set high critical concentrations that result in false susceptible tests. This was something that we wrote. Claudio Cosa actually was the lead author, and it was published last year and showed that a critical concentration of rifampicin, arguably the most significant drug in TB, for 50 years was set too high. And likewise for the other drugs. This actually resulted in a systemic misclassification of resistance, something that we in the microbiology parlance call as very major errors. And why did this happen? Because we actually set clinical breakpoints for TB without clinical outcome studies and without PK/PD. So WHO has actually made amends and lowered the critical concentrations for several drugs, as you can see, the ones in red. This is the Indian programmatic management guidelines. And let me tell you, it is really a good one. We have NAT testing for all TB patients, and we have first-line LPA, which follows at a referral site, which is really good because you're double confirming the NAT and you're looking at INH as well. We've come this far. Do we really need sequencing in TB endemic countries? I'm going to quickly show you a case that came to us in 2014, a 2-year-old little girl who was treated elsewhere for fever, chest infiltrates, who was started on first line, 2 months later presented to Hinduja with drowsiness, intractable seizures, ptosis, miliary TB, and tuberculosis. But she was actually dying. The child was quickly put on IV dexamethasone and anti-epileptics. The child's mother actually, with great persuasion, pulled her X-ray out of her belly and showed that she was on TB treatment, but wasn't improving. She had a dry, non-productive cough and did not consent to a bath. All samples from the baby, GL, CSF, and the mother's sputum didn't give us any joy. And you know, the management in TBM is huge. You have huge dilemmas. There's drug resistance, and in a hotspot like ours, before we up the ante for steroids, we need to know the level of drug resistance. And of course, issues with adequate drug penetration, etc. So how would you treat this sick little baby and her mother without any diagnostic guidance? These are the applications of NGS. I'm quickly going to talk to you about the first one. And imagine our shock when we did pyrosequencing in 2014 and found that this child and her mother both were XDR TB. And believe me, she was spared. They were both spared, giving fluoroquinolone, second-line injectables, ethambutol, pyrazinamide, and ethionamide, which we actually added later. But 4 of the 5 drugs which were ineffective. We put her on a salvage regimen with clofazimine, amoxicillin, linezolid, and PAS. Remember, BDQ and Delaminib were not even thought of in those days. And she was referred to ID for optimizing the treatment, where cycloserine and ethionamide were added. 6 weeks later, the GL and sputum were positive, and we did a LPA, which proved that she was XDR. So basically, we avoided an inappropriate regime with a delay of 7 to 8 weeks and a huge pill burden. Both the mother and child did well at 2-year follow-up. And when the baby came with her mother, 4 years old, to collect her follow-up no growth, I thought this was not just technology. It was the faith of the physicians who believed in the technology who saved that child. Subsequently, we're really finding great use in CSF pyrosequencing based on nucleotide incorporation. Pyrosequencing, we found, is much more sensitive than expert or midget and really facilitates early treatment decisions by detecting, in 6 genes at least 30 SNPs for the 4 drugs that originally defined XDR-TB. We've also asked our program in Mumbai, and we found that we had incremental increase in SNP detection for rifampicin and fluoroquinolones. What about the next? Whole genome sequencing. I'm going to actually only talk on 2 points in this. As we integrated into TB management, one is the transmission dynamics, and we all know that as human beings traveled, traded, crammed into crowded cities, TB went along for the ride. And these are, India has essentially 4 lineages. Why am I telling you this? Because what can happen in a potentially MDR TB hotspot? TB, TB lineages differ in propensity to acquire drug resistance, but the compensatory mutations in MDR overcome the fitness costs that impact growth rate and transmissibility. And with epistasis, these aggressive transmission fit MTB with shorter latency lead to expansion. I mean, look at our crowded trains. You just need overcrowded areas for this to spread as it has. In 2005, we published that in Mumbai, we had a 35% of the Beijing strain. At that time, we thought, and everybody thought, that we had a tertiary referral care bias. Fast forward to 2020, when we found in central Mumbai, a pre-XDR epidemic was being propelled by 3 highly fit Beijing strains with an evolutionary advantage that not only selected for compensatory mutations, but was associated with a higher transmissibility. And this was done by our collaborators in Germany. They showed a 3-fold increase in THD, that is time-scale haplotypic density. The WGS clearly allows for identification of transmission networks with greater resolution. What about what all clinicians long for? This was really a wonderful effort by the WHO, where they actually had a catalog, and India has also pushed out their catalog last year from NIRT. 40,000 strains across the globe were analyzed, and interrogation of resistance conferring variants was found for resistance in 1200. Something we published with the Cryptic group, where we showed that when you had a tier 1 to tier 2, tier 1 is most likely to contain resistance. We could actually get a compendium very easily and look at the, I don't have time to go into this, but this really very beautifully elucidates the number of tier 1 mutations by mutation group in drugs. Remember that the development of this mutation catalog didn't come easy. It was with high quality curated phenotypic data, high quality standardized whole genome sequencing and pipelines. But what was so fantastic about this catalog is that there was a standardized approach for associating variants with phenotypes. Basically, match DST with sequencing data identified to understand the mutations associated with resistance. I'm not going to go into this, but I would advise you to read the catalog because it beautifully describes how matched data identified solo mutations and then went on, and these were evaluated for confidence claim. And look at the way they identified groups 1, 2, 3, 4, and 5, associated resistance, interim, uncertain significance, etc. So the first line drugs, nothing is perfect in life. The first line drugs confidently predict resistance to a really great extent. The second line drugs are far more challenging. For the second line drugs, the new and repurposed drugs, the detection of resistance conferring variants requires molecular interrogation of more than 90 genes. For RIF, we got off easy, just 1 gene. And Public Health England, the Netherlands, and New York are not performing phenotypic DST any longer once WGS predicts it not to have first-line drug resistance. This is a policy change with substantial cost and time-saving benefits. And with GWAS approach, with the Cryptic consortium, we actually found when we included insertions and deletions, we could enhance resistance detection to other drugs as well. Now, there was a lot of controversy. Are the new WHO definitions easy to apply in practice? Yes, it's easy to write out a regimen, but I have a fight to take up with them that the DST has not been looked at. DST for group A drugs is really challenging. Lab capacity for phenotypic DST is currently difficult. Even we find it hard to get BDQ and delaminate. With low resistance prevalence, our PPVs are now going to be good. And sadly, there's incomplete understanding genetic basis of resistance. And this was something that was written by our fine colleagues, which said that the lack of DST for linezolid, bedaquiline, clofazimine, and the delaminate, predominant, and pyrazinamide really is going to compromise our design of the new regimes that we're going to roll out. Luckily, there's a version 2 of the WHO mutation catalog that's going to be awaited, and hopefully we just finished meetings, so it should come out according to Nazir by June. So whole genome sequencing is great, interrogates the whole sequence, but it can currently only be performed on culture isolates. This was a nice study that actually showed the comparison of NGS whole genome with MIGIT. And you can see, I'm not going to go into it, but the red kind of correlate with the red on MIGIT. Here's MIGIT and here's NGS. And if you look at what I always look at in a paper, what were their very major errors? Not bad at all. For MIGIT, they just versus NGS, they just had a 1% chance of missing resistance. Okay. This was another paper that was published, I think, 2 weeks ago, which actually really correlates something that I always look for. And that is, look at this here. It's small. I don't think you'll be able to read it, but it's basically correlating the sensitivity of the high confidence mutations in prediction of drug sensitivity. But better still, it's actually correlating drug mutations for which a dose increase might be considered. That means it's not that bad a mutation. And look at all these mutations for all the drugs here. Why can't it be done directly from sputum? Because human bacteria, viruses, etc., just create a mess. And we actually looked at trying to do whole genome sequencing with MedGenome. It worked a bit, but it took so long that I really don't think it's ready for prime time yet. What about targeted sequencing, where you look at a large number of specified targets adaptable to new genes and is scalable? I call targeted sequencing, pyrosequencing on steroids. It's basically with a faster turnaround time of 2 to 3 days. I'm going to quickly go through this case too. This is a 24-year-old girl who was November last year, severely breathless with cough, emaciation, high-grade fever, and this ghastly X-ray. She had a treatment history, not treated in Bombay, elsewhere, where she was treated with a whole lot of first-line and second-line drugs. And she was so fed up with severe drug adverse side effects, she just started taking BDQ 4 months before she came to us. Her sputum was positive, and the expert, no rocket science here, showed rift resistance detected. We did her LPA, and it showed pre-XDR. That means she had INH, RIF, and fluoroquinolones that were resistant. We considered surgery, but she was simply too sick to take up. So what are the problems with current GST, the genotypic DST that we follow? The repertoire is somewhat limited because for such patients, we need to wait at least 4 to 6 weeks for phenotypic DST to pyrazinamide, moxi, linezolid. Moxi is in high doses, bilaminate, clofazimine, bedaquiline, and also ethambutol and efionamide are not deemed to be reliable on phenotypic DST. Look at the impact of the 2020 new definition. There's so many more mutations. We have to be able to detect all this. So when we did her targeted NGS, we found that she had additional resistant conferring variants to bedaquiline, ethambutol, pyrazinamide, and clofazimine. We didn't find any resistant conferring variants to these drugs. Other drugs is linezolid, etc. And 35 days later, MIGIT bore out exactly the same results. So clearly NGS has an advantage in terms of time. Culture positive plus standard of care gives culture positive plus 10 to 14 days. WGS culture positive plus 2 to 5 days from culture, and targeted NGS directly from sample, 2 to 3 days. We did some work where we did find that in our little setting, we could collaborate with high depth and good coverage. Now we undertook a seek and treat end-to-end diagnostics with Unitaid and conducted by Unitaid sponsored and conducted by FIND. This is up for review. We completed it in December and it's up for review in May 2023, where we looked at 3 platforms. And we actually did 4,000 drug-resistant strains for each of the platforms. Across the world, there were 3 other sites. DPLEX is one of them, which is directly applicable to clinical samples, prediction of resistance to 15 drugs. And you could see this oligotype and identification of more than 100 mycobacterial species. We also looked at version 2 of the MinION, that is the Oxford Nanopore, and found it to be fairly comprehensive, portable, and robust. And we could look at 200 drug resistance mutations. And if you look at it, this is just a chart show across the board where you compare GeneXpert, LPA expert, XDR, pyrosequencing, and targeted, and you'll find that clearly as we go towards the right, things are much, much better. So from a diagnostic perspective, friends, our goal should include the provision of universal DST, not just rifampicin, for all drugs to meet NTB targets. We need to provide the same standard of care for all TB patients, regardless of where they reside in our world. And this is just, I mean, it's shocking. Imagine 2020, 57 of the whole genome publications were from regions that just accounted for 3% of the global burden. We need to wake up. So what ails it? Of course, it's cost and capital investment and high consumables. Of course, it's capacity building and expertise. We need to see how to integrate NGS into our existing diagnostic lab workflows and how to clinically interpret the sequencing data, apart from quality issue, quality control matrices, etc. And of course, decentralization is desperately needed. So why do we need to implement NGS in TB endemic countries? We need to, there is clearly global commitment that is aligned today towards a common goal. And we live in times of unprecedented opportunities. And believe me, we need to build on the momentum to close the window. We need to leverage COVID investments in NGS for TB, as DBT is doing so rightly, quickly. And why? Because new DRTB regimens are going to increase our diagnostic burden. We're going to have to enable, it's going to enable NGS to build a knowledge base and of course, drive utilization, capacity building and implementation diagrams. Is culture-free diagnosis of TB really on the horizon? I think we will succeed with WGS because its surveillance is across the world and WHO especially will largely be based on NGS. But in India, routine surveillance must be continuously done to assess emergence to resistance to the new and repurposed drugs. This NRD is something we need to have a handle on because believe me, it's low now. But before we know it, with the use of various regimens coming up like BPAL-M, we're going to see resistance just insidiously setting in. Targeted NGS has potential to comprehensively diagnose smear-positive MDR-TB with a TAT of 2 to 3 days. Of course, costs will decrease. It's an economics of scale, but we will struggle with possibacillary samples and knowledge gaps in new resistant conferring variants. So as we transform the TB diagnostic gaps, friends, we were waiting for a plug and play, targeted NGS for 2 days, but we will have to wait for WGS clearly and culture for possibacillary. So how close are we? Certainly not today, but we need to jumpstart our planning for tomorrow because ultimately accurate and prompt diagnosis will enable more effective TB control. And yet, sad but true, TB is the perfect expression of an imperfect civilization. These innovations mean nothing if they cannot save lives. Soumya spoke about factors that fuel the ravages of TB that include malnutrition, alcoholism, diabetes, etc., and adverse drug reactions that preclude adherence. Regrettably, this poor girl whom we diagnosed, we got targeted NGS, everything was fine in 2 to 3 days. She was so plagued by her adverse drug reactions, she refused to take her treatment, and I believe she passed away 2 weeks ago. So that's where we are. We need to have, by the time a patient seeks care, look at the whole, we're just a cog in the wheel from all these various maroon circles that you see. It takes long for the patient to get cured. So we need to keep at it because this is all we can do. Thank you.
Thank you so much, Camilla. You put in a lot of information there. I wonder if there are any questions.
We're taking them, Soumya, I think at the end. It's going to be back to back. That's what I was told by Dr. Ramandi.
Okay, so you're going to stay on, right?
Yeah, I will.
So we'll move on to the next. Okay, thank you very much, Camilla. I think we'll move on to our next speaker. There's probably been a slight change in the program. And aap kar lijiye, yes.
The next speaker will be Dr. Anant Mohan, who is a professor and head of pulmonary medicine, critical care, and sleep medicine at AIIMS, New Delhi. He is the chief investigator and co-investigator of several international collaborative and extramural funded projects. And he has worked extensively with respiratory diseases, including tuberculosis, and is a friend of mine. So I'm very happy to have him here. Over to you, Anant, for your talk. Thank you.
And this is amazing.
Thank you very much, sir. Thank you, dear chairpersons. Thank you, Dr. Ramandeep and his team for inviting me here to this very robust scientific session on tuberculosis. So after a very extensive coverage of the lab diagnostics of TB by Dr. Camilla just now, I'll just shift tracks a bit to talk more about the clinical aspects and the therapeutic parts of tuberculosis. Once a diagnosis has been reached, obviously one has to treat the patient. Now, although the treatment is pretty well standardized and fortunately over the last couple of years, we also have some new drugs now with us, but still it is a challenge, and the challenge are at various levels. You know, one is the DOTS program, the procurement and everything program, the program aspects, which are also pretty well standardized. But overall, the treatment is pretty long. Even for drug sensitive, it is 6 months. For MDR, it is anywhere between 1.5 to 2 years. Then there are injectables still a few years back. All this makes treatment difficult, compliance difficult, promotion of more drug resistance. So now in the last 7-8 years, fortunately, we have some new drugs. And what I'll do over the next couple of minutes is take you through the latest drugs which have been discovered and some key regimens and some key trials which have been done and the results of that and how that has impacted the overall management and policy makers. So this shows the overall the 2022 global new TB drug pipeline. And as we can see over here, starting from the discovery phase, preclinical and the clinical phase, and as we move towards the phase 1, 2, and 3, we have these certain drugs coming on, these molecules being tested. And ultimately out of these, based on these trials, which I will show you in some time, the crux of these trials, based on them, some drugs, bedaquiline, delamanid, and pretomanid have been approved now in the last couple of years, and they have been rolled out also in the Indian program as well. So we'll just see how they have impacted the outcomes and how they have impacted the overall regimen. But this just to show that various combinations are now being tried using these new drugs and some other drugs also. These are the 3 new drugs with the regulatory approval, as already mentioned. Just briefly to tell about these drugs and the site of action, just in very brief, as you can see, if we just focus on the key ones, the bedaquiline acts on the ATP synthase. This is the mycolic acid of the cell wall, and then there are other proteins in the various parts of the structure of the organism, cytoplasmic membrane. Over here, pretomanid and delamanid have got multiple modes of action. In the presence of hypoxia, they release nitric oxide. They act on cytochrome oxidase. In the presence of normoxia, again, they may act on the mycolic acid and various other targets as well. Then over here, you have, the other molecules which are still in development phase, and they are also acting on various targets. Won't go into that as of now. Which are the drugs which have advanced into phase 2, and then we come to phase 3. And again, there are a lot of molecules. Even, I don't think there is any point remembering the names, but I just put this slide here because most of you will be a basic sciences audience, so probably the mechanism of action will be of interest to you. So there is one which is an analog of bedaquiline, which has good antimycobacterial activity. And the one concern of QT prolongation with bedaquiline is supposed to be lower with this particular molecule. And then there are others which act on the ATP synthesis, some which act as a beta-lactam compound, which is an antibiotic, some which are similar to linezolid, and the sutezolid is already in clinical trials. Then there are some which inhibit the mycobacterial cell wall synthesis, or other DNA gyrase and other enzymes. Some suppress the protein, and some are similar to clofazimine, and they are being tested as a replacement of clofazimine, which also has got quite a serious side effects again of QT prolongation. This summarizes the various trials of the new drugs which are currently under evaluation. Some of them have already been reported, and as you can see, they are a mixture. Some are working towards reducing the duration of treatment. Some are working towards converting an injectable to a non-injectable all oral type of regimen. So we'll see a few of those subsequently, but here you can see this is a high dose rifampicin type trial in which they have assessed that rifampicin in 35 milligram per kg is also safe and more effective. Then this trial is assessing 6 months of regimen for MDR. The STREAM 2 trial is pretty famous, and that is looking at various combinations in different durations for rifampicin and resistant and MDR TB. And then there are trials which try to reduce the duration from 6 months for drug sensitive to even 4 months of drug sensitive TB is being tried. In truncate TB, actually looking only at a 2-month duration of therapy for drug sensitive TB. And then there are certain others, the next trial, the Xenix trial, some of them we'll just be seeing. So the newer regimens, a couple of regimens are for drug sensitive TB, but the majority will be for the MDR and the rifampicin resistant. This is basically to try and reduce the current 6 months further down to 4-month therapy using rifampicin. And if we just briefly see the drug design, this is the control arm, which is the standard which is being given. This is, mind you, this is only 2 drugs in the continuation phase. On the other hand, there is a rifampicin, which is given in for 2 months first, and then for 2+2 over here. And rifampicin along with moxiflox and other drugs remain the same. So this again, a total of 4-month therapy, again a 2-month for this control arm, and then a follow-up of around 18 months. The primary endpoint as being assessed at 1 year. What they found was here that the combination of rifampicin with moxiflox was non-inferior to the current standard 6-month regimen. And this was assessed by means of a negative smear and culture, and there was no increased incidence of any major adverse events. So this one compared with this one was non-inferior. Most of these trials are non-inferiority trials rather than superiority trials because they are comparing against a particular existing standard of care. Then the truncate TB trial, which we had just seen, this also looked at a controlled standard treatment, a 2-week standard treatment followed by a HR therapy, and they had 4 comparators, 4 different regimens of comparators. Again, all of them are only a 2-month therapy, so 8 weeks, all having different combinations. Again, the baseline therapy of HZE was there in all, as you can see in all of them here. And the add-on therapy was a high-dose rifampicin linezolid, high-dose rifampicin clofazimine, or with rifampicin and linezolid, or bedaquiline and linezolid. So these were the 4 combinations which were tried. Now, unfortunately, 2 of them stopped the recruitment because it could not be completed, and these 2 are the ones which have been continued. And what was found was the 8-week bedaquiline linezolid regimen again was non-inferior to the standard. So this one was as good as this one over here, with a total shorter duration of therapy and no safety concerns. So here also they kind of say that, a 2-month therapy containing bedaquiline. And mind you, this is bedaquiline for drug sensitive, not for drug resistant tuberculosis. This is not something which right now in India is being advocated. Then there are other ongoing drug sensitive TB new regimen studies. One is the Simplicity TB group, which also looked at a 4-month regimen comprising bedaquiline based moxiflox, parazinamide. Then there are RifaShort and Redefine studies, which are looking at higher doses of rifampicin. So attempt to cut down the duration translates into increasing the dose of rifampicin. So higher the dose, probably we can give shorter regimen. That is the kind of hypothesis on which all these trials are being done. This was the WHO drug-resistant TB regimen till 2019. After that, the new one has just come in 2022, and that we'll be seeing later on. And here, of course, you can see that the all-oral regimen is the one which is containing bedaquiline. Over here, the shorter regimen is containing an injectable. So the injectable was also something that was, one was trying to do away with. So what were the trials that replaced injectables? And this was the Stream 2 trial, and one is the next trial. What is the Stream 2? Here, there are basically 4 kind of regimens. One is a 6-month regimen versus a 9-month all-oral regimen versus a 9-month regimen. Again, this contains injectable here versus the standard of care, which was the existing 20-month WHO regimen at that point of time, which was then terminated because the things changed. The outcome, very, very briefly, the outcome was that both the bedaquiline containing regimens were superior compared to a 9-month injectable containing regimen. And there were obviously fewer cases of hearing loss because that is associated with the injectable, the amikacins or the kanamycins. Another trial was the next trial, which compared a 6 to 9-month therapy containing bedaquiline versus the injectable regimen. Here also, they found a favorable outcome in favor of the bedaquiline containing regimen of 51% versus almost 20 to 23%. Adverse events were also more favorable towards the bedaquiline containing regimen. A word about the Stream 2 trial. The Stream 2 trial was published just a couple of years ago, and this had four regimens comparing with each other. Long one around 20 months, which was the one which was existing at that time, at that point of time in the WHO. Control regimen contained total of 40 weeks with a 16-week of intensive phase, and you can see the drugs which are being used over here. So intensive phase had the injectables, but not so after that. Then one was the oral regimen, in which there was no injections, but it was a total 40-week therapy. And then there was a truncated 6-month therapy, in which again there was an injectable 8-week intensive phase. So this is how it panned out, regimen A, B, C, and D. And based on this, what they found again was that both the bedaquiline containing regimens were superior compared to the 9-month injectable containing regimen. Again, the hearing loss was a concern always, and that was also less. So basically the bedaquiline did better as compared to the injectables at that point of time. Very recently, the next trial was also, the results are out. It's a phase 2-3 combined open labeled RCT, which evaluates 6 to 9 months of an all oral regimen. And you can see it is a bedaquiline and linezolid containing regimen versus a standard of care, which was the 9-month WHO injectable based regimen. Again, they found that the all oral 6-month regimen had a significantly improved outcome, clinical outcome compared to the injectable. And you can see it was almost 51% versus 23%. When bedaquiline based therapy became the standard, this was conducted in South Africa. So when the bedaquiline based therapy became the standard of care, so the trial had to be stopped prematurely because the experimental arm itself became almost like a standard of care. Then there are certain trials which not only replace the injectables, but also they shorten the duration. Both kind of outcomes were looked at. And these are the 3 or 4 trials which we'll just see very briefly. The NICS-TB trial looked at XDR and MDR-TB that were not responsive. And this was basically the BPAL regimen, which was a 6-month regimen at that time. Then there is a Xenix trial, which looked at different doses and durations of linezolid. And we look at that. We had already talked about the Simplicity, the TB trial. The drug sensitive we have seen, there was an arm of drug resistant TB as well. And then there was a TB practical trial in which they compared the 6-month BPAL versus a BPAL M versus a BPAL clofazimine versus the standard of care arms. The NICS-TB trial, it was basically on 109 patients there, compared a 3-drug regimen containing the bedaquiline, pretomanid, and linezolid versus the standard of care, looking at clinical resolution at 6 months after therapy. And almost 90% of all the patients had favorable outcomes, and the outcomes were pretty much extremely good, and that made the world take notice. They did find some peripheral neuropathy and myelosuppression in a significant extent in the linezolid containing group, but this kind of a response in MDR-TB was hardly ever seen before. Then the step 2 was to try and see what is the dose of linezolid that would be best. And there in the Xenix trial, they looked at 4 different type of doses of linezolid: high dose 1200, that is for a longer period, 1200 for a shorter period, 600 for a longer period, 600 for a shorter period. And based on that, the bedaquiline dose also was slightly modified here. It was changed to a daily dosing just to improve the adherence. Otherwise, till that time, they were giving it alternate day. What they found over here was the favorable outcome was seen in all groups. So these are the 4 groups. Favorable outcome was seen actually in all the groups. Least was in this one, but otherwise it was more or less 90 to 93%. Peripheral neuropathy was there almost in 13 to 38%. Myelosuppression was there. Some linezolid dose modification was also required. But overall, so like when we see the efficacy range from 84 to 93, but overall, when they looked at the side effect profile, along with the benefit of response, it was found that the regimen of linezolid in 600 milligram for 26 weeks actually was considered to be the best. So out of all these 4, that seemed to work the best with the best safety and the best efficacy. Then came the TB practical trial, which was a 6-month all oral regimen for these drugs. Here what they did was, this is the BPAL plus M, and this is the BPAL plus clofazimine, and this is BPAL, and this is the control regimen, shorter as well as the longer regimen. And this is how it panned out. At 6 weeks, at 16 weeks, they changed the dose of linezolid, And what they found was that all the investigational groups met the eligibility criteria for progression, but ultimately the steering committee only proceeded with the BPAL-M group over here. They had a good favorable outcome, the BPAL-M, again 89% versus 52% of the standard of care. It was actually terminated because efficacy was so good after recruitment of 75% of that. And mostly because of treatment-related adverse events in the other group, in the standard care group. So basically, this also showed that the BPAL and the BPAL-M regimen really worked very well. There is some evidence and some studies from India also, and one of this, that is a Beat India study, which is a prospective open label cohort study in pre-XDR of 165 patients, where they combined these drugs for a total of 24 to 36 weeks, and they found a favorable outcome in more than 90% of these patients. There was quite a bit of myelosuppression and peripheral neuropathy, but minimal cardiotoxicity and other myelosuppression, etc. While detected, it was treated successfully. So this is something that was also back home. This is an important study, although not a proper RCT, but still gave a good bit of information here. The WHO guidelines on TB very recently have suggested basically, again, remember, these are all conditional recommendations with very low certainty of evidence as of now, and probably evidence is going to build up, but whatever was available, based on that, they have suggested the following: a 6-month therapy comparing, that is basically a BPAL regimen, rather than a 9 or a longer month therapy as was being advocated before. And they also suggested a 9-month all oral regimen rather than an 18-month regimen in patients with MDR, where resistance to fluoroquinolones was excluded. So these are the two key take-home messages from the WHO right now. We have the programmatic ones from our 2021, and probably we will be modifying or maybe adding up to that as well. Maybe some updates will be coming out very soon. So just I'll conclude with this, that the biggest challenges to the treatment of TB actually for us actually remains compliance. Diagnostic, of course, extremely important, but once that is done, it is the compliance to therapy, especially to the long therapy, and the emergence of drug resistance because of incomplete therapy and interruption of therapy. But now, fortunately, newer drugs and newer regimens have enabled significant reduction of the duration of the therapy now of the drug-resistant TB from the earlier 2 years, almost 2 years. Now, shifting from the injectable to the all-oral regimen definitely is going to go a long way in ensuring compliance and patient adherence. So that is definitely a welcome step. And we do hope that based on the emerging evidence, short course all-oral regimens could also, if things go well, could become the standard of care in managing DRTB in the years to come. Thank you very much.
Thank you, Anand, for an excellent talk detailing all the chemotherapeutic regimens that we have for drug-sensitive and resistant tuberculosis. Can we have questions now? Or maybe you can join us here and then we can have question after the last talk. So it's my pleasure to invite the next speaker, Dr. Karan Madar, a friend of mine, again, additional professor in the Department of Pulmonary Medicine at AIIMS, New Delhi. He has been working on interventional pulmonology, sarcoidosis, tuberculosis, and many other diseases, and has received multiple awards for his excellent clinical research. So he's going to talk about mediastinal tuberculosis, which is obviously a diagnostic and management challenge. Over to you, Karan.
Thank you, sir, for your kind words. It's a pleasure to be here, and thank you, Dr. Ramandeep and the entire team for inviting me to be a part of this gathering. I was quite unsure that what will be the kind of audience I'll be speaking to, so I probably thought, and I think I've done the right thing. Probably it's a lot of students who are here. So I think I've tried to keep my talk a little simple to give you a basic overview of what we are talking about, and to tell you about a specific subgroup of intrathoracic tuberculosis, which is different from pulmonary tuberculosis. And why is it important? Why is it challenging? And what are the research areas that this topic can get us into? So let me begin by putting this cartoon in front of you to, show you what we talk by the mediastinum. We all see images of the chest or the thorax, but it's a big compartment. And for everyone, and we talk about tuberculosis, we presume that all intrathoracic TB is pulmonary tuberculosis. Pulmonary tuberculosis meaning that your lungs are actually getting involved by tuberculosis. But when we talk about mediastinal TB, mediastinum is the space which is lying between the two lungs. And it actually houses the heart, the great vessels, and also it houses a lot of mediastinal lymph node or glandular structures inside the mediastinum. And in some patients, the involvement of tuberculosis can actually spare the lungs, but have an involvement in between the lungs. So that is basically what we mean by mediastinum. So it's a pretty extensive area, but what it actually tells you is that these are the airway structures which are close to the trachea and the bronchi. And these lymph nodes can be extensively involving, from the top to the bottom. So basically a large, chunk of the surrounding the airway structures have these lymph nodes, and these can get involved with tuberculosis. So why is it important? Because a fair proportion of TB in India is extrapulmonary. Among extrapulmonary TB, the lymph node is the most common site, but this mediastinal lymph node TB is a separate subset of extrathoracic lymph node TB. And it is difficult to diagnose because of a lack of clinical examination findings. The symptoms can be very non-specific. The X-rays can be, eventually normal in a large proportion of patients. And the sputum examination can be non-yielding because there is no communication with the tracheobronchial tree. So this is outside the lung parenchyma and the airways. So if you see the chest radiographs, in pulmonary TB, you'll see, the involvement of the lung parenchyma. In mediastinal TB, what we are talking is about the central area. And these things can be missed by, untrained radiologists. And, if you see this X-ray, it seems nearly okay. But, the small bulge which points towards a lymph node enlargement, these things are frequently missed. So that is why this mediastinal TB is not picked up. And X-ray is a very, very insensitive tool to pick up this condition. Now, if you talk about, what tells you that there may be an involvement of the mediastinal nodes is a CT scan, thereby bringing us to the thing that, CT scan facilities are also not available, very freely across the peripheral, areas. And what you get is these lymph nodes which are here. And what points towards TB is, necrotic appearing lymph nodes, because necrosis is a sign of tuberculosis. But what makes this thing more challenging is that a fair proportion of patients do not show necrosis on the CT scans. So thereby telling us that clinically also it's difficult to pick up, and on imaging also it's difficult to pick up. So now how do you, get to the diagnosis of a lymph node involvement which is away from the lungs? And classically, once we talk about TB diagnosis, we talk about either getting acid-fast bacilli or culture positivity or CBNAT or other molecular tests, or you combine a clinical radiological profile along with a tissue sample and response to treatment. And in the context of TB, extrapulmonary TB especially, you need a histological evidence of either a necrotizing granulomatous inflammation with or without acid-fast bacilli positivity. Now, another important feature is that a fair proportion of patients with TB can have a non-necrotizing granulomatous inflammation also. So how do you get the tissue, because this mediastinum is not in communication with the lungs and the airways. The sputum will be non-yielding in a majority of patients. And these lymph nodes are, usually also beyond the scope of bronchoscopic visualization. Sometimes we do a bronchoscopic procedure in which, a scope is put through the airways to examine the airways, but these structures are outside of the airway. So a routine bronchoscopy will also not, give you a visualization and a diagnosis. So that is why you need advanced diagnostic modalities to go beyond the airways. And that is why the diagnosis becomes a little more challenging. So what you actually need to do is actually do something to puncture beyond the airway, thereby something called as a transbronchial needle aspiration. So this is a modality in which, we all hear about bronchoalveolar lavage. You've heard about BAL samples. BAL samples means you just go inside with a bronchoscope, put some fluid, take the fluid out. But this will not work if you're talking about something which is outside the airway. So what you do is actually take a needle and you puncture it, and try to get the sample. Now, what I've shown you on the left side is something which is called as a blind puncture, in which you don't know, you know where the anatomy is, but you puncture that side. But now what has revolutionized the understanding of mediastinal TB is the advent of modalities in which you have a guidance on your bronchoscopes. So what you call something as an EBUS, which is an endobronchial ultrasound. That is ultrasound inside your airways, which actually allows you to pinpoint the lymph node and do a real-time aspiration. So the supposedly inaccessible site of intrathoracic TB is now accessible for diagnosis. So this is what a blind puncture looks like, which is called as a conventional transbronchial needle aspiration, or also called as blind, because you are actually seeing where you are puncturing, but you don't know what is beyond that. So that is the blind part. This is how it looks like. These are the mediastinal nodes you can puncture. And actually, this is how this is a small, this is a cartoon to actually give you a real feel how it looks like. So, you know, this is a carina which divides the right and left bronchi. And herein you can see that the needle is used to puncture the, you know, airway wall, and you can take samples. So this is what an EBUS looks like. You have a dedicated bronchoscope, which allows a needle to go into the airway, and you can visualize the needle in real time. So it's important to understand these are complicated procedures because these require a fair bit of sedation. The procedures are longer, and actually this also entails a bit of risk more than a conventional bronchoscopy. So that is why mediastinal TB is a little challenging to diagnose. And I'll also be coming to the other important point that even if you get a tissue, why is it difficult to diagnose? So this is actually a small cartoon to show you what it looks like. So what you get is a view of the lymph node glands under vision. And if I just freeze it here, you can actually see needles going in real time into the lymph nodes. So you actually know you are taking out the material which you need. So this is how you get the tissue samples from the lymph nodes. You get these cores of tissues and also the cytopathological material. So giving you an idea of somebody who's in research, interested in research in this area, that what are the kind of samples one is dealing with. So there is a very sparse data, good quality data on the clinical profile of mediastinal TB also. But this is a work from our center in which I've brought out a few salient features to tell you that why it complicates the matters. Tuberculin positivity is seen in around 78% of the patient. This means if you just go by a tuberculin positivity, that is not good enough for giving you a diagnosis. Fever is not a very common symptom. 40% of these patients don't have fever. Nearly 1/4 have normal chest radiographs. And the heterogeneous or a necrotic appearance of a lymph node on a CT is also nearly half of the patients. So that is by meaning that if you do an X-ray, you do a CT scan, then also you are not clear whether you are dealing with intrathoracic TB or not. And the gene expert, even if you get a sample, is positive in only 40% of the patients. So you get a tissue, but is that enough? So that's the important thing. You get a granuloma in the sample, but is that TB? So the problem is that we have also a very major intrathoracic condition in our country, which is called as sarcoidosis. Previously, we used to think that sarcoidosis does not exist in India. But in our work, in our center, the most of the endobronchial ultrasound procedures that we do, the most important indication is sarcoidosis and lung cancer and then TB. So what is this condition? This is a closed TB mimic. So what you have, it has also got granulomas, classically non-necrotizing granulomas, and it's a very common indication of evolution of mediastinal lymphadenopathy in India and a very close mimic of mediastinal TB. So we recently published a fairly large experience from India of the clinical profile of more than 300 patients with histopathologically confirmed sarcoidosis. And I'll just bring some salient features which are important, that a small percentage of patients with sarcoidosis can have necrotic lymph nodes. And very interestingly, we found that around 13% of the patients with sarcoidosis have a necrotizing granuloma. So that is why, getting a necrotizing granuloma in a sample does not make it tuberculosis. And we also found, this is not described before, that a 10% positivity, on tuberculin samples, whereas it is believed that if you have a positive MON2, it cannot be sarcoidosis. So what I'm trying to bring to it is that we have two common conditions in our country, sarcoidosis and TB, which share very common and overlapping clinical features. Even with extensive evaluation, which is very complex, the differentiation between these two entities can be challenging. So once coming to treatment, there are challenges because we don't really know, we don't have good quality data on how long to treat mediastinal TB. Most of this is from extrapolation from lymph node TB and pulmonary TB, in which a standard 6 months anti-TB regimen is given. But if you follow up these patients, in many of these patients, the lymph glands will persist despite treatment. And we don't know that a persisting node is inactive or active, and we don't have markers to tell us whether the disease is active. So regarding the therapeutics also, this is a challenging area. So what are the research implications? This is a audience, which is very, focused on basic research. So the bottom line is that you see what a complicated course one has to go through to diagnose that mediastinal TB. You see, at a peripheral setting, you don't have even a CT scan. Even if you have a CT scan, you don't know. Then you have to refer the patient for a complex bronchoscopic procedure, which will be only available in a tier 3 city or an advanced facility, and that also will be expensive and complex. So can we find relatively lesser non-invasive methods to diagnose mediastinal TB? Can we find something to differentiate TB and sarcoidosis better in our country? Because we have a fair bit of sarcoidosis with us. So it's not all granulomas are not TB. So how do you differentiate them? How do you do studies to study the efficacy of therapeutic response in a non-invasive manner, and development of diagnostic biomarkers for granulomatous mediastinal lymphadenopathy? So summarizing, mediastinal lymph node TB is a distinct entity within the realm of intrathoracic TB. So all the TB inside in the thoracic cavity does not mean we are dealing with pulmonary TB. We have a subset of extrapulmonary TB inside the thorax also. Mediastinal TB and sarcoidosis share overlapping clinical features, and they are often very challenging to differentiate even when you have performed the sampling. And nowadays we have the availability of bronchoscopic modalities which are fairly advanced, that allow a confident diagnosis in most patients with granulomatous mediastinal lymphadenopathy. The advantage of these modalities is that if you do studies using these advanced diagnostic modalities, your gold standard diagnosis is more certain. You are not talking about just a clinico-radiological diagnosis, and using these group of patients as your gold standard, you can actually work towards non-invasive diagnostic modalities to find the diagnosis of these entities. Thank you very much.
Thank you, Dr. Madan. Can you join us? So fantastic talk, and he highlighted that how the PPD in about 7 to 8% of cases of tuberculosis can miss. And on the other hand, about 10% of the sarcoidosis cases also can have PPD positive. So this is one thing. Another thing which is, he said, rather emphasized, that necrosis, which is a, we consider hallmark of tuberculosis, that is also sometimes found in, sarcoidosis. But most importantly, for a clinical scientist, what he emphasized, that his study and AIMS study shows the dogma in what we are taught in medicine, that sarcoidosis is not a problem in India. But what his study shows that yes, if you look it, you will definitely find. So I think with that, both the Dr. Mohan and Dr. Madan, both are here to have some questions there.
Camilla is also online, so we can direct questions to her as well. Questions from the floor, please. Yes. Lady at the back, yes.
I have a question for Dr. Madan. So you were saying that people who have mediastinal TB do not have fever, and they do not also have other symptoms. So when do they actually come? Because if they are going to a normal physician, X-ray will not show that they have TB, pulmonary TB. Their other tests will probably also not show. So when do they actually come to you?
So that's a good question. So a fair bit of mediastinal TB which we diagnosed was incidental also. So, or with patients with minimal symptoms who are complaining of dull aching chest pain. Some just got radiographs just like that. and they were detected to have mediastinal glands on the CT scan. The problem happens that if you have detected a node on a scan, you need to confirm what is it. So that is why, you can have very non-specific symptoms also. I said that the fever was there in 40% of our patients, thereby meaning they won't have fever. But this means that they will have also symptoms apart from fever. Some may have a dry cough, some may have a dull aching chest pain. Interestingly, we had patients who came with a primary ocular presentation. Patients had a uveitis, blurring of vision and a red eye. On a CT scan, you found they have just lymph nodes and they are diagnosed as TB. So I think what it means is that the presentation can be quite varied, but how you pick it up is on a CT scan and definitely diagnosis when you sample the lymph.
Okay, sir. Thank you.
Yeah, I also have a question here. My question is a follow-up to this about mediastinal therapy. So as you see that the numbers are fewer and difficult to diagnose, so do we have like the cases where positive cases and their radiographs or CT scans? If that can be fed, and perhaps the AI-based diagnostics can be done based on all that we know, so that it will be non-invasive and perhaps it will also add to your research, plus maybe, who knows, it can be a diagnostics also.
Yes, a point very well. So that's what I mentioned. See, the diagnostic algorithm for diagnosing mediastinal lymph nodes is not straightforward. It requires a bit of complex procedures. So anything or any research which can aid to a non-invasive diagnosis is most welcome. So what you're talking about is a radiological differentiation between these two entities. We could also have biomarker-based studies to differentiate those entities, but the problem is that the sample size that you would need to do such a study with biopsy confirmed cases is the challenge. So I think this calls for more multi-center and collaborative work to, get a larger sample pool of gold standard diagnosis patients and then work on this topic.
Bill Bishai from Johns Hopkins. Thank you, Dr. Mohan, for your wonderful presentation. In the United States, we have way more M. avium and M. abscessus pulmonary disease, and we routinely use inhaled amikacin. Why has inhaled amikacin lacked investigation for MDR and XDR TB?
Yeah, so inhaled antibiotics here are mostly used for critical care settings sometimes. It is true that it is not one of the standards of care for MDR TB. There were some very initial reports. I would say probably something about bioavailability might have something to do with it initially, because drug delivery systems at that point of time by the inhaled mechanism were not standardized. They are even not standardized now. But in the last couple of years, the emphasis has been more on oral therapy, especially in India. One other thing is that, the program, from the programmatic point of view, public health point of view, giving tablets is much more simpler and all, rather than giving inhaler or inhaled therapy. So that again, operationally, I think if we look at it, it becomes a challenge. That is why probably it is not really merited, but I am not able to give you an exact answer how much amount of work has been gone into it, but not the standard of care. Yeah.
So I have the last question for Dr. Rodriguez. Do you think in near future we will have targeted sequencing for MDR, XDR tuberculosis coming to the clinic?
Yes, I think so, because once WHO endorses it, I think the programs can then start. These people who have a MySeq or an iSeq or even a nanopore, they can invest in it. Once the endorsement comes with a rigorous STAG committee review of the study that was just done, I think we'll be in a better position. And for pre-XDR, I really feel there is a role for this, because we're treating in the dark. And in another 2-3 years, when we start seeing resistance to bedaquiline, delamanid, and linezolid, clofazimine, which is already there, in our work, we found it to be about 3 to 6% for all the 3 drugs. I think we need to have a handle on upfront, because these are seriously ill patients. And I always say that for MDR TB, and both the clinicians will bear me out, you have just one chance. You can't mess it up. And then later on try to. I think it will come. It will take time for us to sort of, develop and teach But there is a huge scope for it, I think, and we must do it.
Okay, thank you very much. I'd like to thank all the speakers and the chairpersons for this session. We are moving on with the next talk, which will be online. Over to you, Krish.
Now I humbly request Professor Rajesh S. Gokhale, who's currently the Secretary for the Department of Biotechnology, Government of India, to deliver his keynote address. The thematic focus of his research group has been to elucidate the complex interplay between metabolic reprogramming and immunity in the context of pathogenic disease, tuberculosis, and autoimmune skin disorder, vitiligo. He is also the recipient of Shanti Swarup Bhatnagar Prize for Science and Technology, one of the highest Indian science awards for his contribution to biological sciences. He was the former director of the Institute of Genomics and Integrative Biology. Sir, we request you to deliver the keynote address.
Thank you very much. Can you hear me? Can you see the slides? Thank you very much, all of you, for giving me this opportunity. Can you hear me?
Yes, we can hear you and see the slides. Over to you.
Okay, thank you. Thank you very much. Well, I think, first of all, let me thank you, all of you, for giving me this opportunity to speak at this meeting. Incidentally, I am at the G20 meeting in Dibrugarh, and I thought I will just, for a very short while, just join and say hello to all of you. There are lots of friends from my own TB fraternity whom I've had cherished wonderful memories with. Thank you very much again for coming to this important meeting. And I think, let me first of all, Department of Biotechnology actually has initiated a program on INTGS, which is basically got to do with the genome sequencing of large number of genome sequencing. And they wanted me to inaugurate this logo of the INTGS and the website. So I would first do that part of my work. So this is the new logo for the INTGS, which is an Indian Tuberculosis Genomic Surveillance Consortium, a program that is actually based on the INSA-COG activities, which is actually a SARS-CoV-2 genome surveillance activity that we have going on. So we're building on those activities. And our whole idea really is to actually make sure that similar kind of genome sequencing programs actually drive Mycobacterium tuberculosis understanding in future as we move along. This is the new website that just been launched actually, and I think it will be accessible for everybody to be able to look at. The whole idea really is to, in about a year and a half, to genome sequence about 30,000 strains. In collaboration, this program is done in collaboration with Health Ministry, ICMR, CSIR, several of what you would call a full government approach actually. And I think many of these samples will be strains collections will be deposited at one place and should be accessible to everybody to use, and make the best out of it in future to come. I'm sure this will have tremendous amount of impact as we go along into our research activities of tuberculosis in various forms of what we want to do. Just to give you a perspective, I mean, the kind of data that will be present over here, as you can see, this is a very early data from about not more than, I think maybe about 180 genome sequences that have been completed in last week. Just to give you the present status of the resistance that you can identify in all these things. And I think clearly the problem is very, very important, therefore, that we actually map out our own genome sequences, understand the resistance patterns that we are going to be circulating around in the tuberculosis strains as we move along. Let me go back to my talk that I was originally had wanted to give. This is, I thought I'll talk about the nutritional economics of tuberculosis actually. And I wanted to bring a couple of perspective, particularly from a perspective of Indian community to bring to the forth and how we can address these problems. I think, as you all know, treating tuberculosis is thought to be pura course pakka ilaj. That is, the whole idea really is that directly observed treatment, which is defined as short course, and the short course is 6 months. Actually, unfortunately, it's not that short, is really the patient-centered case management approach that WHO recommended and has been implemented for almost last more than a couple of decades actually in India. And clearly there is obviously a challenge, as several of you were discussing, but just before I joined for a short while, I could hear several interesting discussions with regard to multi-drug treatment and other mycobacterial infections that Bill was referring to about actually. And I think the question is that why, despite 3 decades of implementation of this multi-drug regimen, we still have about 4.5 million die every year from what you would call a treatable disease actually. And primarily the reason for this that is given is actually patient non-compliance to drug therapy actually. And I think obviously this is one of the major reasons. However, the emergence of drug resistance forms due to a variety of other issues also could be a major challenge. And we need to address all of these problems as we move into it. I think there are 3 important questions that sometimes I like to ponder about from the NTB game. Poverty and poor nutrition are classically associated risk, as I said. India had committed for NTB by 2025, a rather ambitious goal from a government perspective, but I think it's important to set difficult timelines so that people actually try and bring themselves up to the task. So it's very, very important to do that. And I think it's probably the whole idea really is to create momentum and make sure that many good things happen in a short period of time. And obviously the 3rd pillar of NTB strategy, we need to intensify research and innovation. So the questions that I've tried to put together actually that I keep worrying about from the point of view of at what age group TB is prevalent in this country and in general in the world as well. Is there any change in disease dynamics in recent years, and are there therapeutic regimen associated side effects due to long course of treatment? And long, which you mean by shorter course of treatment. I think one of the very interesting aspects that you realize is that about 33% of the total TB cases, TB burden in the country has a age range of 15 and 30 actually. And classically, you know, the 2 questions of is this an age to have weaker immunity is kind of questionable to look at. And also inadequate nutrition, because this is supposed to be the age where you have maximum amount of energy in your life, is also something that one needs to look at. So there must be a non-optimal performance index that one needs to look at and be able to carry out this activity. Other than that, one of the major impact over the 19th century, you know, the consumption began with the cough actually. And I think this used to be tuberculosis is another name called consumption and was used to be characterized as fatigue, excessive weight loss, and general wasting away of the victim actually. And so also the component of undernutrition, which has actually been, taken up in various contexts actually, that over 40% of youngsters receive less food than they should. And therefore the malnourishment is thought to be one of the major challenges, despite the fact that the GDP of the country has gone, the quintile of incomes between various families, between lowest and highest, is very large. And therefore, we still consider this as a very, very important activity. And I would think so. This is a very important factor to look at. However, by treating tuberculosis, as you can see, this is a medical prescription that's given in India. You can see that there is an antimicrobial drugs, which is typical of all. And at the same time, micronutrient supplements of various kinds are given actually. Now, there are various ways in which supplements actually recover and help TB subjects. But what is also important to understand that how many of them recover and how many of them are able to bring, or does these micronutrient supplementation works across the different kind of zones or across different people, or is it restricted to certain people and not to others? And are we missing something in terms of their treatment protocol regimens actually? In India, as you would see, Nikshay Poshan Yojana, which was initiated actually a while ago in April 2018, in fact, provides a financial incentive of about 500 rupees per month to each notified TB subject. And this is done through direct benefit transfer and has been given to 9.25 lakh patients adopted for nutritional support as of now. So it's a fantastic initiative if it's, and should bring value in future as we look at actually. However, there are challenges and problems in terms of infection, immunity, and nutrition as well. So for several years, this aspect of TB has been rather very poorly thought to, even though, as some of you would remember, the TNF-alpha, which is a classical cytokine associated with TB, was used to be called cachectic factor. The nutritional partitioning during treatment of TB is in fact rather, not very well understood. And I think what has been suggested for some of the early work is that typically the fat mass is regained, but the protein mass is not regained actually. And you can see the P ratio, which is what talks about the protein ratio of content of the body, in fact, changes after the initial part of the treatment, but doesn't recover fully to the complete levels that normal people should have actually. And therefore, there are these kind of problems that the quality of life actually just become a challenging. And therefore, the question that we should also start asking now that is very important, is that whether the TB subjects are actually can we bring them back in a complete physical and mental health, such that they can be back to the work and do what they're important to do. What is also interesting to look at is that because of vast epidemic of pandemic and impact of COVID-19, actually there are a large number of interesting immune related activities that have come up actually. And the large number of cases which are long-term effects of COVID-19 that have been discussed very widely. In fact, we don't end up discussing MTB long-term effect that widely actually. So if you look at this, is a diagram that I picked up from one of the paper, and it talks about immune dysregulation, microbiota dysbiosis, autoimmunity and immune priming, blood clotting and endothelial abnormalities, and neurological abnormalities. And many of these are listed pretty heavily, in very large number of patients. And fatigue, which is one of the major symptoms to look at, which is about 58% of people who have long-term COVID is affected by it. So I think it's all telling you some interesting perspective of how infectious diseases in fact has a very important host component, and it's the recovery and the quality of life of the host component should be also a very important parameter as we look into the importance of tuberculosis recovery patients actually. So the risk factors for incidence of TB individuals actually, as all of you would know, and I won't go into details of this slide, but just to that COVID-19 pandemic has further exacerbated the situation by threatening the financial security of socioeconomically vulnerable individuals, and also has disrupted food disruption networks. And therefore, it is very important that good nutrition as well as various benefits of prevention of complications are provided at the right period of time. Several studies as part of the TB report cohort has been done, and they provide some interesting perspective, and probably Dr. Sonali is there. She may talk about some of these in her discussion actually. But what is important to look at, the severe undernutrition at a treatment initiation and several severe undernutrition before the onset of TB were both associated with unfavorable treatment outcomes actually. Now, the concept that one needs to talk about is that whether the severe undernutrition at treatment initiation and lack of BMI increase during treatment are also associated with 4 to 5-fold higher unfavorable TB treatment outcomes actually. So I thought I will talk to you about some of the work related to this aspect that we did in our lab. But I think this is an important area to look at of nutrition, immunity, intersection, and how the infection and how the recovery of quality of life is so critical actually in terms of lung function, in terms of hepatic functions are critical, such that the younger population that is impacted by TB actually in fact have a very good quality of life in future to look in future part of their work. So concept that I thought I'll talk to you about is about nutritional immunity paradox. And again, a very, very important aspect about how metal ions that are actually that are typically not produced in the body but acquired. And so there is always a competition between host and pathogen to acquire these metal ions, iron, zinc, or copper. And on the left-hand side, you can see what happens is that there is a concept of starvation-mediated bacterial killing, whereas there is on the right side, there is a context where is poisoning-mediated bacterial killing. So these are two contrasting features, whereas in one case, we are talking about that metal ions are sequestered or effluxed out from the site, such that the pathogen is not able to utilize these, and therefore host can in fact make pathogens weaker and thus can be cleared out. On the other side, in fact, host also utilizes an enhanced level of these metal ions to in fact do the poisoning impact as well. And these are kind of rather interesting mechanisms by which the host is able to manage the pathogen differentially. And it's not yet clear how these two extreme host responses are tackled in the body. So in my lab, we've been interested in looking at zinc metallostasis as host pathogen interface. And there are some interesting studies that are starting to look at. So what we wanted to do was we wanted to, under the normal zinc condition in the normal cultures, which is about 6 micromolar zinc, and the low zinc and high zinc, we adapted the strains and we wanted to understand what is the difference between that low zinc at the optimal zinc and at the high zinc. And the optimal zinc is the one that is there in the cultures of MTB that we utilize. And what is the difference between their growth and content of zinc content, or even in the case of their ability to, what are the genes that are upregulated that we can identify or altered, which makes an impactful this. So if you can look at the growth are not very different. In fact, they are very similar to each other. There are no significant changes. And so there are not very many significant changes that you observe between these three sites, even in the content of zinc that is present inside the bacteria for a certain number of generations of cycles actually. So clearly there is a mechanism by which the host metallostasis is maintained, host pathogen metallostasis is maintained. Now this is the data that, this is a plot that probably gives you a perspective of some of the key genes that are modulated during these two extreme conditions of zinc. And you can see the pathway that emerges has got to do with the efflux pathway, with this ESX pathway is actually known to be involved in various activities of pathogenesis. But the other one of the interesting pathway that also emerges is got to do with the cluster, with the 10 kb cluster that has been known as an NRP cluster for several years and was identified and is one of the largest cluster that is present in Mycobacterium tuberculosis. You can see that in both under low zinc and high zinc, this cluster that consists of 0097 to 101, and in fact, probably 96 is also part of it actually. All of them go up around in both these conditions. And the question that we are asking is why under low zinc and high zinc does this cluster in fact go up, and what is the role to play in terms of zinc metallostasis? So this is the 11 kb operon, as I said, and has enzymes and a non-ribosomal peptide synthetases, which is a bimodular enzyme, non-ribosomal peptide synthetases that can activate 2 amino acids along with acyl carrier protein, fatty acyl AMP ligases, a thioesterase-like protein, and an oxidase actually. So what is also unique about this cluster is that this cluster is only present in pathogenic species, and from all non-pathogenic species, this cluster is absent and obviously has been therefore for several years talked about as one of the key cluster involved in pathogenesis and virulence. And large number of studies over the years has in fact been demonstrated to show importance of this cluster using various mechanisms of transposition mechanism, proteomics, biochemical assay, overexpression in various conditions to form metabolites, and also deletion, and trying to showcase various role in biofilm formation and all. However, the molecule that is produced by this cluster has been unknown. And we wanted to understand whether this cluster is really, what does this metabolize is made? And can we understand its functional importance such that we can correlate molecule function and its physiological significance to look at? So what we were able to do in our lab was to actually do retrobiosynthetic approach, isolate these proteins, characterize these proteins, and being able to identify a putative molecule that would be produced by this cluster. And you can see this is a dipeptide that is acylated to a nitrogen of a what we first initially thought lysine, but it turned out it's an ornithine, that is a residue that is present here. And so it's a phenylalanine ornithine, and the two long acyl chains that are produced by a rather interesting pathway where fatty acyl AMP ligase activates a AMP form fatty acid, loads onto an acyl carrier protein, and then a thioesterase again across the alpha beta unsaturated fatty acid adds a glycine molecule. And this then glycine molecule is oxidoreductase enzyme converts into an isonitrile. And this chain of isonitrile containing lipid chain is transacylated on these two amino groups to make this interesting lipopeptide. And this lipopeptide is then released by a reductive domain as a phenylalanol. And therefore, this molecule, which consists of phenylalanine, ornithine, and long chain acyl, is what we called actually as a Kupia-4. So this you could identify by doing mass spec based analysis, followed by chemical synthesis, and followed by careful fragmentation patterns to be able to demonstrate that in all these class of molecules that are produced contains a common MSMS fragment, which consists of a core of dipeptide, and then various fragments confirm their masses that are present actually to be able to look at. So it's so we also synthesize this molecule, then amino derivative of this, which could be converted to by, and then we use this isolated metabolite converted into an amino derivative, and then we could demonstrate on an HPLC that it has an identical mass and an identical molecular weight, confirming that the identified molecule is the one that we have predicted to be able to look at actually. So we call this diacylated diisonitrile lipopeptides. Actually also was recently reported in a earlier work in M. marinum, where where the isonitrile lipopeptide contain a single amino acid. And we call these molecules as Kupia-4 because of their rare metal capability to carry forward lipid as a carrier of metals. So we wanted to ask this question, what's the importance of this molecule during the infection cycle, and when is it produced in mice context? So therefore, the typical mice model was used, and actually you can see what you can clearly see is that the mutant that does not produce this, which is a delta NRPS, is actually fairly compromised in its early stage of infection, and in the later stages probably appears to be catching up, which was also reported, as I showed you in my other slide, by other people as well, actually. But it was interesting to note that the Kupia-4s are in fact only produced during the early part of infection, and they probably disappear by the day 21 of the infection cycle, actually. In fact, the cluster gets completely shut off, and we are now identifying a pathway by which these molecules get degraded actively, such that the host does not utilize these molecules any further for doing other activities. We could also identify these molecules in the freshly isolated cultures of TB infected the clinical strains that we identified from various patients, actually kindly provided by CMC Vellore. And you can see in all of these, one can identify these molecules in different form, clearly indicating that as you do clinical passage, these molecules are lost because you can't identify these molecules in the culture conditions, for example, H37RV or strains that are passaged for a longer period of time. So we had proposed a possible model where we proposed that these molecules are produced inside the cytoplasm and then they secrete it out, and then they go out in the milieu, and in fact capture zinc and also probably copper, and they bring back this into the cell where they now are kind of released, and then this Kupia-4 is recycled back into the system actually, such that providing a mechanism of maintaining a kind of an important role in the TB physiology. So similar metallophores for iron has been identified and has been known since 1965 in Mycobacterium tuberculosis, and it's interesting that it took about almost more than 50 years to identify molecules that are involved in zinc and copper metallophosasis. So the question that we are trying to address now in the lab, and is what is more interesting, is that what we realize is that our Kupia-4s MTB mediators of systemic zinc dyshomeostasis that are leading to TB, leading to cachexia actually. And the reason we believe that some of these molecules are very important for the work, because primarily much of this cachexia work is going on in the area of cancer. And what you mean by it's a multifactorial, as you would know, cachexia is a multifactorial syndrome involving ongoing loss of skeletal muscles that cannot be reversed by conventional nutritional supports and lead to progressive functional impairment. And there are papers that have shown that the ZIP14, which is a critical mediator of cancer-induced cachexia, in fact, redistributes the zinc to the muscles and therefore result in cachectic, one of the possible mechanism by which cachexia occurs in the muscles of the patients actually. It'll be worthwhile to look at and see whether something similar happens in some of the patients whom even despite giving zinc supplementations or other supplementations are not able to recover their lung diaphragm functions or cardiac muscle functions. And therefore, it's going to be important to understand how these complex mechanisms in physiological context and in the context of inflammation and nutrition can in fact function well and be able to carry these activities. I think it'll be, that's going to be the future of my lab that we are looking at right now. And some of the students, I think Kriti, who's done much of the work, is actually there in the meeting, maybe can tell you a little bit more about it. But it's about really what we are starting to more and more understand, that even infectious disease is all about interorgan crosstalk and how the inflammation and the tumorokines and the metabolic mayhem that is set up in the case of cancer, in fact, gets triggered in several complex diseases that survive for various longer period of time. And because of this complexity, one needs to address this in a rather holistic perspective, rather than limiting itself to the pathogen disappearance as the only way of treatment of TB actually. And I think I will end here by acknowledging several people who wonderfully in a very short period of time were able to contribute to this work. Actually, Dr. Srinivas Reddy did the synthesis work. Dr. Siddesh Kamath did all the fantastic mass spec work. Guru Prasad, who is there helped us with the zinc related activity. Dr. Mohanty did the computational work. Farooq did the RNA-seq work, and Dr. Joy was a clinical partner, and Tanya Parish and Anil Oja helped with the mutant strains actually, and all the students who have done fantastic job. And thank you very much for giving me this time and opportunity. And I'm missing all the good things over there. I'm sure that is going to happen along with the meeting, but I hope I will be able to see you all, and some of you stay back. I'll be happy to meet you on Sunday. Thank you very much. And I will stop sharing my slide actually. If there are any questions or otherwise.
Thank you, Dr. Gokhale, for an excellent talk as usual, but this time more science oriented rather than policy oriented. So it's a very nice shift from policy to science. Any burning questions? Otherwise, we can end the session here. I think there are no questions here because it's a keynote lecture, so we generally don't have questions. So thank you very much for this wonderful talk once again.
Thank you very much. Thank you. Thank you.
Bye.
I think I just wish to say hello to all my friends and several good friends there. Thank you very much for coming, and sorry I would come back and see you actually
some other time
at the G20. Thank you. Bye.
Bye.
Thank you very much, sir. I thank all the speakers and the session chairs for this very exciting and informative session. I request the session chairs to kindly felicitate the speakers. Dr. Sarman Singh to felicitate Dr. Anant Mohan. Dr. Pramod Garg to felicitate Dr. Karan Madan. I now request Dr. Jayanta Bhattacharya to felicitate the session chairs, Dr. Pramod Garg and Dr. Sarban Singh. Yes. Thank you, sir. Thank you, everyone. We now break for a very quick tea. The tea is arranged in the second dome area that is to the right of you. I request everyone to assemble back in the auditorium by 5 p.m. for the next session.
पकड़ो ये कौन सा है सेशन 2
We'll start the program in 10 minutes.
Hello. I request all the participants who are outside in the dome areas to immediately come back into the auditorium. The program is going to start in a moment. Thank you.
Welcome back, everyone. After a very exciting and informative session one and some brainstorming over tea, now we begin with our keynote lecture titled Discovery of Bedaquiline and Aspirations for Shorter New Pan-TB Drug Regimens by Dr. Anil Kaul, who is currently the Vice President and Head of Discovery Research at the Global Public Health R&D Unit of Johnson & Johnson and a Professor of Translation Discovery at London School of Hygiene and Tropical Medicine. Dr. Kaul's key achievement is the discovery and development of bedaquiline. Dr. Kaul has been awarded the Heroes of Chemistry Award, the Sun Pharma Research Award, and the prestigious French Prix Galien Award. He is currently a core member of the United Nations Top TB Partnerships Working Group on new TB drugs. Following the keynote address, there will be a 5 minutes question and answer session. In the interest of time, I request the audience to keep your questions brief so that there's opportunity for others. Sir, I request you to please come on the dais to deliver the keynote address.
So good evening to everybody. And thanks organizers for giving me this opportunity to be here to talk. And I'm so excited to be here. And the excitement is, of course, because I know so many people here. The people I grew up in the same labs, like Nishit here, Ramandeep here, and also my mentor, Professor Anil Tyagi is here also. So it's such a memorable experience, and also a lot of my CSIR colleagues and colleagues from the other Indian institutions. So it has been, I have been looking forward to this opportunity to talk to all of you. So of course, my talk will be focused on bedaquiline and aspirations for shorter new pan-TB regimens. Today, we heard a lot about it in terms of fantastic talk by Professor Mohan when he talked about the new regimens which are coming for TB. So TB, I can tell you that is in terms of innovation, I think this is the best phase for TB R&D across the globe with the new drugs, new trials, new diagnostics, potentially new vaccines, and as Soumya was also talking about, and the new RNA therapeutics. So this is the outline of my presentation, introduction to bedaquiline. This year, we will have 10-year anniversary of bedaquiline approval globally after the FDA approval. So I will mostly talk today, not much on the science part of it. I will talk about the impact, what a discovery can have in terms of the impact on the patients. And also I will talk a little bit towards the end, some new targets, some new biology, which we are trying to do, and my own inspiration of world without TB. So as you saw from my affiliation, I am part of, I have two affiliations. I have a lab at London School of Hygiene and Tropical Medicine. And I also have a discovery R&D group at Johnson & Johnson in terms of what we call global public health. So we are a big group where we work on 4 major pillars. One is tuberculosis and leprosy. So TB is, I keep my passion alive, or I keep myself alive because I work on TB. So TB is something which we have a huge program on TB. Then we also focus on dengue and flaviviruses. So we here focus on dengue prophylaxis and dengue treatment using small molecule approaches. And we also have a program like everybody else, every institution, every organization works on coronavirus these days. So we thought that let's not stay behind. We also have a very nice program on new target, new biology, new chemistry on coronaviruses. And finally, AMR, which nobody unfortunately works on. But there is something the world needs to do and take it more seriously, because AMR is going to be the next TB. So with all what is on the plate, how do we do it? Of course, we are a very small team. We are extremely focused. So when I was in India, I realized how much public health institutions across the world have the value. They have so much infrastructure, so much talent, and so much of fantastic science happening in those institutions. So what we did is that we created this framework of what we call Centers for Global Health Discovery. So these are the labs which we operate at other public health institutions in the world. Like we have recently signed an agreement with Duke NUS, where we are focusing on dengue and flavivirus R&D, because they have extensive expertise in us, and the Singapore government is really, really focused on trying to mitigate the impact of dengue in Singapore. And in University of Cape Town, we are looking at AMR, because AMR is coming up more challenge in low and middle income countries, especially the African continent. At London School, we have a lab which is completely focused on TB exploratory R&D, which we normally don't do in the companies. So really the aim has been to fill in the innovation gap. I can give you just one example in terms of the dengue, because the story is new. It came last week in Nature, where we showed that, or the team showed that basically there's a compound called JNJ 1802, is very effective in preventing dengue replication in the monkey model. So very nice data showing that pre-exposure prophylaxis study. So this is the design of this particular study, where you have monkeys which get infected on day 0, but on day -1, you start the treatment of this particular compound once daily up to day 10, and by day 28, you terminate the study. So just a small glimpse that this is the vehicle only. This is the untreated monkeys, and you can see that the viral kinetics is very nice of the dengue virus. And this is the low dose, 0.01 milligrams per kg per day dose. And you see that you start seeing some difference in the kinetics at the low dose also. At the mid dose also, you once again see that delayed viral replication in these monkeys. And finally, at high dose, you see this red line here is a complete suppression of the viral replication. So amazing data in terms of the efficacy. And the dose is also very nice, because 3 milligrams per kg per day is an extremely low dose. So what the group did is also they defined how does this particular compound act. So it acts by a completely novel mechanism of action, which is blocking this protein called NS4B. So NS4B has to interact with NS5, and they form a part of the replication complex. So this is the replication complex in the Golgi apparatus, when the virus is being assembled together. And blocking this interaction between NS4 and NS5 basically prevents the dengue replication and prevents the assembly of the virus. So really important data, not only from the science perspective, but with the hope that one day this molecule will be the first therapeutic which will be able to treat the dengue infections across the world, treat or even prevent for that matter. So let's go to bedaquiline. I think a lot of you know that this particular study where we have had this drug, and this is called diarylquinoline, and it impacts the ATP synthase. And this is the C subunit of ATP synthase, and it basically binds it here. And what was very important from this particular study was that we at that time didn't understand the importance of this particular number. That is the impact on blocking the enzyme function of the ATP synthase with such a low, such a high potency and such a high selectivity also. So it came out the IC50 was almost 2.5 nanomolar. What's also important is the IC50 and MIC correlation between on Mycobacterium tuberculosis. So this was one important aspect of the whole, what we call target identification and validation strategy. But what is also important, which we didn't realize at that time, we were so unaware of the whole thing at that time, that this particular, we had the activity on the replicating bacilli or the MBCs, not MICs, MBCs. So you can see the MBC of rifampicin is so potent, while MBC of bedaquiline is not as potent as rifampicin or isoniazid. But when you look at the vein-sidal concentration, which is the activity on non-replicating bacilli, and then suddenly you see the impact that you have very nice activity on the dormant bacilli, as good as activity on the MBCs. And the ratio of WCC to the MBCs was really nice. As compared to rifampicin, it loses the activity on the vein-sidal bacteria as compared to isoniazid, which is completely inactive. So no understanding of what it does mean in terms of the activity in mice or activity in humans for that matter. But then subsequently we showed that indeed this activity is very, very critical for showing the sterilizing potential of drugs in the mouse model, as well as efficacy. Eventually, you have seen today how much bedaquiline is used in clinical studies. So this is a paper which was published last year by a group in Canada, where they showed for the first time the cryo-EM structure of the entire ATP synthase. So it was, of course, M. smegmatis, and I think the video will work. So if you haven't seen it, it's a group, very fantastic work done by this Canadian group, where they showed the structure of this is the C subunit of the ATP synthase. This is bedaquiline molecule coming in, and it prevents the interaction between the A subunit and the C subunit. So the interaction between these two subunits is very, very important in order to basically transfer the proton from A to C. And if you block that transfer of the proton from A to C, it basically kills it stops the function of the whole enzyme. And you can see that each monomer here, initially we thought that it was only one molecule of bedaquiline binding to the whole oligomeric ring. But now what they showed that each monomer actually has one single molecule of bedaquiline across all the C9 ring. This is a ring. You can see that all these molecules of bedaquiline are out there. Really amazing work in terms of defining the specificity of this drug towards this particular target. And also the implication, there's a lot of data in this Nature paper where they showed that the implication in terms of the torque which this enzyme creates and has an impact on the molecule. So once again, another interesting study which we did after we had a very weak first clinical study we did in South Africa, where we showed that the drug was not really active when we gave it to patients for 7 days. So nothing really happened, or very weak. I'm not showing you those data, but not really impactful. But then there's one of our own friends from Dr. Tyagi's lab, Neeraj, did this particular study where he showed that indeed, when he did this single cell time-lapse video microscopy, nothing was happening during the first 7 days. This is the time in days. This is the CFU, and you can see that the curve is really flat. But after 7 days, it really started having the impact. So you start to see the kill. And this is reflected in this particular video, where he labeled Mycobacterium tuberculosis with the GFP. And then for the first 3 days, you see the growth. Then after 3 to 6 days, you start adding the drug. And then from day 7, you start monitoring what really happens. So this is an experiment which was done for almost 27 days. And you can see that how exactly this, it's not working, but there's a very nice video. You can see it online also, where you find the impact of the drug over a course of time. But this was really classical study, which helped us basically say, okay, now instead of 6 days, we can do a 2-month study. So that's why it's very important to have very fundamental understanding of the biology. So it's so critical to help and do some critical studies as we go into the clinics. So this was the first study which was done in a 2-month trial by this guy, Andreas Diakon. And he instead of 7 days, now we said, let's do it for 2 months and let's see what is the impact, whether it works or not. And to the whole surprise of everybody, this is in MDR TB patients which get ethanolamide, pyrazinamide, ofloxacin, kanamycin, and on top of that, this is the arm which is basically where you get the bedaquiline. It was called TMC 207 at that time. And this is on y-axis, it's a culture positivity of sputum. And you can see that after 2 months of the treatment, you had 50% of the patients achieved cultural negativity. That means there were no bacteria in the sputum, while only 10% on 5-drug background regimen, showing definitely one of the amazing data I had seen in the history of the whole program. Of course, now, since then, a lot of studies have been done, as Professor Mohan talked about. This particular study, this is the Lancet 2018 study, where they did a meta-analysis of almost 12,000 patients across 25 countries in more than 50 studies. And what they found after this meta-analysis is that bedaquiline was really almost 88% had a success rate, and around 12% we had failure or relapse rates with the bedaquiline. So really impressive in terms of what we call real world evidence, not a clinical evidence. So this is also very important. He did briefly mention it, but I really want to re-emphasize here is this particular study, what we call NICS-TB study. It's basically a study done in South Africa where bedaquiline was combined with proteanimide linezolid, and after 6 months, they saw what really happens based in the patient based on what kind of resistant bacteria they have. So XDR TB, as you know, it's one of the hardest to treat. It was one of the nightmare because every XDR patient had a chance of almost 20 to 30% to live. 70% times they will likely die before bedaquiline and the other therapies came in. So, but now with this particular combination, you can see that almost 90% chance of increased survival. And it was as good as what we see in the MDR TB population. So introduction of new drugs does definitely improve your mortality and also improve the chance of survival. So now coming to another important aspect is the resistance. So we talked about resistance. Camelia talked about how NGS is shaping the DST is so critical to understand the resistance. But we also need to understand the molecular basis of resistance. How does resistance emerge? So in this study, we looked at the efflux pumps. So if you can see the efflux pumps, you have two kinds of efflux pumps, which are, some of them are ATP driven, and some of them are driven by what we call a proton motive force. So bedaquiline, as we know, the major impact is on the ATP production. So if you look at this particular ATP production, you definitely see that if you decrease the ATP of mycobacterial cells, it will have impact of other processes which need the ATP. And efflux is one of those processes. And in the same way, proton motive force is very critical driver of efflux of some of these PMF-driven efflux pumps. So you have these here. So what we, at that time, we didn't realize the importance of this particular study. We did this microarray. We saw that the moment you put bedaquiline on these mycobacterial cells, you have down regulation of efflux activity. You can see it here, you can see it here, and these are all efflux pumps listed here. But that's an experimental observation. How do you connect this experimental observation to a clinical observation? So this is basically data which is already out there in public, where we show that bedaquiline reduces the risk of pre-XDR, XDR, and new resistance to other background drugs. So this is bedaquiline group, where only 2 out of 66 patients developed new resistance. And if bedaquiline was not there, the placebo group, you had almost 16 out of 66 patients developing resistance. So that means bedaquiline does prevent emergence of resistance in the background regimen. Whether it is related to the efflux pumps or is there something else here, we don't know. I think we don't have experimental evidence right now to showcase that. Interestingly, a lot is talked about these days on this RV0678. So when we first did this particular experiment, we simply identified mutations in this particular. First, we found it highly upregulated. We didn't understand what the upregulation means, but over a period of time, we found that this upregulation was associated also with, in certain cases, with the mutations in this particular pump, in this particular protein. And eventually what we looked at, this was a transcriptional repressor of this particular operon, which codes for MMPS5 and MMPL5. So this actually, these two proteins form a efflux channel. And the moment you have a mutation here, there is no repression, the more efflux protein is formed. And the moment you have more efflux protein, of course, it leads to gradual increase in the MICs. And now this is the data which is coming from 2022 New England Journal of Medicine paper. They show bedaquiline resistant tuberculosis with RV0678 mutations. So this does tell you the importance of the understanding the intrinsic as well as target-based resistant mechanisms. At the same time, what we had done in this DREAM study, we looked at almost 5,000 MDR-TB isolates in the treatment-naive patients and looked at the resistance pattern. So these were pre-XDR or XDR-TB isolates. And overall, what we saw, which is nice to hear, that we had a resistance emergence between 1.5 and 2% in this particular patient population. Now, different people have different reports, but the baseline resistance was between 1.2 and 2%. So just to give you the final picture of this particular section, we have high lipophilic drug, bedaquiline. For those who are in the chemistry here, they will understand this a little bit more. This is the PCA plot looking at all the antibacterials, and the red one are TB drugs. If somebody tells you I can design a new TB drug, I think it's considering this, it's very hard to understand what exactly properties we are looking for new TB drugs. Of course, ATP synthase was not target. It was not validated. Everybody criticized it. And the effect on human mitochondrial ATP synthase, that was an issue, and delayed bactericidal activity. I talked about the experiment using the time-lapse microscopy. Also, the sputum culture conversion. So the clinicians don't see this as an endpoint. It's always a surrogate endpoint. The endpoint is the relapse at that time. Nowadays, everybody uses a sputum conversion as a clinical endpoint. But at that time, we were told that it has to be a relapse study. The elimination half-life is really long for this particular drug, and that for some people that was an issue. So the key is that in spite of all the challenges, in spite of all the issues, in spite of so many drawbacks and failures also, but sometimes you have to be lucky. Let's be honest, you have to be luck on your hand. But at the same time, it's very important to keep on doing things, even if the odds are against you. So you keep on pushing the envelope so that you reach to a stage what you are looking for. So the impact. Now, as I said that we are 10 years up, so let's see the impact. What does this particular molecule and what it has done in the field? I think the first and foremost, I don't know whether you realized it when Professor Mohan said that now we have an all oral regimen. All oral regimen means that now these patients don't need to go to the hospital every day for 6 months to get an injection. So for me, that's the biggest achievement for any new TB therapy. So I think that has been a transformational impact, that the pain and the misery of these patients to go every day and have a 3 ml injection. This is the first drug with a novel mechanism of action. More than 500,000 courses have been given. 158 countries have access to the drug, so which is also very good. 72 countries have regulatory approvals, and we have the medicine Betaclin is on WHO's list of essential medicine for adults as well as pediatrics. So let me tell you, because this is a translational science institution, you will understand it. Discovering drugs is sometimes very easy, or innovations are easy in the labs, and a lot of us try, but to bring it to the patients, what we call access, which Soumya was talking about in the morning, is one of the most difficult things to do, because you have to work with regulators, you have to work with governments, you have to bring the drug. Professor Sarman will know it, in the programmatic framework of the countries, you have to build those supply chains. It's a very, very complex thing. So I think the last mile approach is very, very critical for any new innovation to succeed. So that's why it's interesting. What's also interesting is the pediatric TB. Not many people talk about children. And Soumya said this is so much neglected because nobody cares about it. So we have almost 1 million cases of TB in children every year. A lot of them are MDR patients. So what we did is that, which was very nice in a sense, that we were able to not to use just break the table and give it to kids. We were able to develop a dispersible formulation of Betaclin, and after doing clinical studies and using what we call weight or age-based dosing, we were able to identify 20 milligram formulation of the drug. And you can see that its palatability is very important for kids, adherence is very important, and sputum conversion, of course, is very key. So eventually the drug got a pediatric formulation, was approved by FDA in 2020. So leaving aside TB, I think some of you who know the genetics of Mycobacterium leprae know how leprae is compromised, how so many genes are deleted, and how the whole genome is basically shrinked. So what we did is that basically there are certain genes like cytochrome BD and few other genes which are not out there, and the MIC of Mycobacterium leprae the MIC of Betaclin on Mycobacterium leprae is really good. So we thought that, okay, let's see what really happens in patients. So we took these Mycobacterium leprae patients, looked at y-axis is the HSP18 expression from the skin lesions, and on x-axis you have treatment weeks. So you have week 8 here. So you can see by week 8 we had complete eradication, not eradication, we had complete negative signal of the PCR in these patients. So let me tell you, leprosy is the oldest disease in the world. We have 200,000 infections still happening every year. So it is definitely a burden. And with my hope is that with this particular drug, and of course in a combination therapy over a period of time, a phase 3 clinical study, this is just a single study, it doesn't say much, you will be able to have an impact on the on Mycobacterium leprae and maybe hopefully eradicate it. So what next? Next, of course, we have a lot of TB researchers here, a lot of people working on new drug discovery. So what we need is drugs which are better bactericidal activity, and maybe also sterilizing. And as we saw, some of the studies, the push towards shortening the treatment duration. Can we shorten the treatment duration to 3 months, less than? My goal is, if you tell me, can we shorten the treatment duration to less than 3 months? But Professor Mohan showed that there are some studies which are trying to go to 2 months also, bedaquiline and linezolid in normal TB patients. It's sometimes very risky because you have the risk of resistance coming through. Of course, oral is very, very important, that it has to be oral, safe with HIV. It has to be universal. It doesn't need the DST. As Camilla was saying, the complexity of these treatments, you have to have the NGS sequencing done and this done, and everything has to be matching. So can we just simply get 3 or 4 new drugs and combine them together and make a universal regimen? And of course, better compliance. It has to be compliant, able to comply. I think eventually that's one aspect of it, combination of 3 or 4 drugs. Can you bring all these 4 drugs into one single pill? One single pill for treatment of TB universally and less than 3 months. That will be fantastic. That's how the future of TB should look like. And it's not easy because of this. You have such a difficult what we call heterogeneity of TB in patients. This is major human pulmonary TB lesions. As was said earlier, here in the clinical section, you have non-necrotizing granulomas, necrotizing granulomas, fibrotic granulomas, different types of granulomas, which don't allow the drugs to get in. The fibrotic, there is nothing gets inside. So how will you treat these patients? So then, of course, based on that, you have heterogeneous TB populations which are sometimes replicative, sometimes non-replicative, sometimes intracellular, sometimes extracellular, sometimes have low nutrient availability, sometimes have low oxygen tension. So it changes the dynamics, how you can, what kind of a drug you need in what kind of a physiological state of the TB. So yeah, I will quickly finish it. So apart from the impact of what all the assays, what we do, this is some of the assays we routinely run in our. But apart from assays, we really need to embrace new technology in terms of the drug discovery. And one of the technologies which we routinely use is what we, and nowadays everybody uses, the AI/ML platform. So we have an AI/ML platform where we do mostly the medicinal chemistry design. So we design new molecules, taking different elements, but we also have what we call high content imaging platforms, where we can see the image of every molecule, impact of every molecule on a mycobacterial cell or a eukaryotic cell, and image it. Impact on nucleus shrinking, impact of cytoplasm, impact on so many mitochondrial biogenesis and things like that. You can really do it in a single experiment. This is respiratory chain of TB. You all know it very good. This is the ATP synthase. My hope is that we can still deliver on cytochrome BC, cytochrome BD, menaquinone, purine biosynthesis, work in this space and make 3 or 4 drugs combine here and make that one pill fixed dose regimen, the pill I showed you. It's not easy. So why is not easy? If you look at BC inhibition, that BC is this particular target listed here. If you look at BC inhibitors, when we compare these inhibitors on H37RV versus clinical isolates, it's very interesting finding. So you see that on H37RV, there is very small kill, but when you have clinical isolates, you have very, very nice kill. So sometimes what we do with the H37RV in the labs may not be reflective of what is happening in clinical strains. So indeed, then if you move the same study, what you do in vitro, in vivo, you see the H37RV, what we routinely use, and we have this particular clinical isolates in the mice. And this is the BC inhibitor, which we used here. Not much major impact. But when you compare it in the clinical isolate, you definitely have a bigger impact. So once again, heterogeneity, the genetics of the strains and everything what you use has an impact on the drug delivery. Menaquinone is something, as I told you, very interesting. It's absent in humans. It's essential in replicating and non-replicating thing. And also, there is also the purine biosynthesis, which we are working on. What we have in menaquinone is compounds which have very nice IC50s on the enzyme, nice MICs, very extremely potent molecules. And for purine, we have identified something which is extremely bactericidal, so bactericidal, much better than anything I have seen. But the question always remains with these metabolic targets is how relevant they are and in what physiological conditions you have done your assays and what is your measuring background. So a lot of still unknowns and understanding which we are completely lacking at times. This is one of our visits to South Africa, where we went to a MDR TB clinic. I keep on showing this image as a reminder to everybody that this is a pharmacy in South Africa, and the BL is basically the boxes of Bedeclin. So it was very nice surprise to see all the boxes there, because this reminded us every day that whatever we do has an impact somewhere you don't even know or you don't even connect to them. Before Bedeclin was in this clinic, almost 70% of XDR patients, if this was a XDR or MDR TB center, had a very low survival rates. But upon Bedeclin approval, we had a very fantastic thing. This is a tweet also correlating to this by Ministry of Health and Government of India, where they in 2019 rolled out Bedeclin across more than 500 MDR TB states in India. I think this is the last slide. It says it all starts with the discovery. A lot of us are discovery scientists. So I really, really believe in the power of discovery, innovation, science, and every discovery what we do in the labs, whether it is translational, non-translational, it has an impact. Science in general has an impact. If somebody tells you that translational science is better than basic science, that's all, let me not use the word, but it's whatever we do has fundamental impact. And I don't want to read it out loudly, but you can read from this particular quote. I liked it. He tweeted it recently, but the essence of this quote tells you all that what we need to do for TB or science in general. So with that, I would like to thank you and happy to take questions.
Thank you very much, sir, for walking us through the journey of discovery of Bedequilin and shedding light on avenues to shorten TB regimen. I request Dr. Shinjini Bhatnagar to felicitate Dr. Ranil Kaul.
Maybe we can leave the felicitation. Let's have questions.
Any questions? We have some time for 5 questions. Yeah, please.
Why is it delayed kinetics for 6-7 days? I'm sure you've done so much of work. It should be fairly easy to figure it out.
I think if you tell me from a molecular level,
yeah,
I don't know.
Okay.
I have
no
understanding.
Did you look at the pumps during that period? Maybe at that time, because ATP level is good enough, and the molecule itself is thrown out of the cell.
Maybe that's a good.
So that is one
yeah.
clue. Second, what about linalosid? One of the antacids, not the first line, but maybe second, third line. That also inhibits ATP synthase. Okay. I'm sure that paper came in PLOS Medicine in 2012 or 15, around that time. And right now I'm not able to, but he's a very famous person. He's the one who first time sequenced MTB. He was there, Stuart Cole. He's the one who he reported that. But it was not pentaparazole, but one of the derivatives of it. I think something starts with L. So I think there is already a molecule existing which inhibits ATP synthase, originally described for inhibiting malaria growth. Same molecule, that is ATP. I have another question that is in relation to dengue. You talked about prophylaxis. What about if you give it after a delay of a day?
You caught me.
I will do it.
No, indeed. I think after you give the treatment, it depends upon the treatment window. Because of all, whether it is COVID, flu, RSV, and dengue, the treatment window of viral replication is very, very short. So you have to come at the right treatment opportunity. If you come between the first 28 hours in the flu also, we have seen that in patients, unless until you don't come in the first 28 hours, no treatment works. You have to be at the right time immediately after detection. The same thing is with the dengue. You have to have an early intervention in order to have an impact. If you come when the virus is and decline, you don't see any of the impact.
Okay. I have one more, but later.
Anil, nice talk. So my point here is, I want to talk to you about your point of bedaquiline attacking the ATP synthesis, right? So coming to the host side, so if you block the ATP synthesis, you are going to arrest the macrophages, especially CD14 and CD68, and inhibit their phagocytic activity. So how, did you ever come across any kind of immune polarization status of the patient who have received this bedaquiline? I'm just asking for the let's say prospective point of view, whether bedaquiline will also polarize the immune system while taking care of the mycobacteria in the host.
Yeah, to be honest, I don't know what immune polarization means in the host. In humans, I have no idea what exactly that means, but I can tell you that we have done the impact on the human ATP synthase and the in vitro assays, and we don't see impact on the ATP synthase function. So there is the catalytic binding site of mycobacterial ATP synthase versus the human ATP synthase is somewhat different. So that's what drives the specificity, not only for TB, but for other bacteria like E. coli, staph, or all the other bacteria which also have the ATP synthesis.
Dr. Kaul, it was indeed nice talk. So there are some reports mentioning about the side effect of bedaquiline. Do you have any comments on that?
Oh yeah, there are. Every drug has a lot of side effects. So one of the major side effects when we were developing was the QT prolongation. And that's the QT is the heart, and you have the QT wave. It gets prolonged by 20 milliseconds. So that was one of the side effects. But as we have rolled out and done a lot of clinical studies, so that has no impact on any adverse events. So we don't see any. Of course, then you have nausea, you have stomach aches, and a few other things which are associated with a lot of these drugs.
I just want to ask, how do you see bedaquiline working with INH? Because INH works on bacteria which has higher respiration rate. And if bedaquiline is known to decrease the respiration rate, do you see bedaquiline working with INH?
Oh yeah, that's a very nice question. INH is very active on replicating bacilli. Bedaquiline is active on replicating, semi-replicating, and dormant. So you need combination. In TB regimen, what you need is a combination of multiple drugs acting on different heterogeneous populations. And yeah, you can really have a very nice combination of INH and rifampicin. Of course, bedaquiline is not approved for the first line TB therapy. So right now that combination is not used anywhere in the clinics.
Thanks, Dr. Kaul. Certainly, we all agree the bedaquiline has changed the way TB is treated today and in near future. The only question I have, keeping in mind, somebody asked the heart, sorry, one of the ways was to conserve the drug. Because we have a drug after 50 years, we conserve it for future more and more as far as possible and use in more critical cases. But I see, and as a company will definitely do that, I appreciate, is to make use into many other combinations, pediatric and this and that. So finally, the bottom line is that the drug exposure in the community is going to increase every day. Are you thinking of preserving it in some way that it is used widely, yet you could preserve it for much more longer time?
Oh yeah, that's a very important question in terms of antibiotics. Every antibiotic leads to a resistance if you don't use it properly. I think we misuse antibiotics too much. And the same thing is here. If you put it in the front line, it will be gone after some time. So you have to have strategies, controlled access, monitored access. And that's why I think WHO and every national TB program is very concerned that we don't misuse the drugs, particularly bedaquiline. And that's why it's only right now approved for drug-resistant TB, where it is needed the most. So it's as a second line drug, it's not at the primary first line. So it's very important that you create that framework of cautious use of antibiotics, and that includes bedaquiline or any other drug, anti-infective.
I have a question on bedaquiline. So given that its mechanism of action is on ATP synthase, and we look at the said negative effect in terms of similarity to mitochondrial ATPase, but can there be a positive effect also, given that some of the disorders may require the downing of ATP synthesis, like ischemia or strokes? So it could have a protective effect also. So have there been any studies where bedaquiline, besides treating TB, has a protective effect?
Yeah, I think there are some studies around it. There are some publications of use of bedaquiline in cancer patients and certain malignancies. But these are very individual reports of trying to see the impact there. So, but there are some reports out there.
One last question. No, this is regarding little bit of cardiotoxicity associated with the molecule. Do you think it is? Do you think it is more to do with the individuals already having abnormal QT wave? You see, a small percentage of patients undergoing.
Yeah, no, that's not the case, because when you do a clinical study, you monitor the baseline, and then you see the increase above the baseline. So the baseline is there, and then you see the increase. Whether there is a genetic susceptibility to increase QT, that we don't know.
Sure, a lot of questions, and Dr. Anil is going to be here for the next 2 days. You can catch him whenever you want. Thank you very much.
Thank you very much, sir, for walking us through the journey of discovery of bedaquiline and shedding light on avenues to shorten TB regimen. I request Dr. Shinjini Bhatnagar to felicitate Dr. Anil Kaul. Thank you very much, sir and ma'am. I request you both to be seated on the dais for chairing session 2. For this session, we also have Professor Shinjini Bhatnagar as the chair. Dr. Shinjini Bhatnagar, currently the Professor of Eminence at THSTI, has been heading the maternal and child health research at THSTI over the last decade. She was the former Dean for Clinical Research at THSTI. She comes with an experience of 25 years as a pediatric gastroenterologist at the AIIMS Delhi before joining THSTI. Professor Bhatnagar is also associated with the Ministry of Health and Family Welfare, the Indian Council of Medical Research, and the Bill and Melinda Gates Foundation. Dr. Bhatnagar is a fellow of the Indian National Science Academy, the National Academy of Medical Sciences, and the National Academy of Sciences. We have 5 talks in this session, each for 20 minutes. Following the talks, there will be a brief question and answer session with all 5 speakers. I request the audience to hold their questions until then. I urge the speakers to stick to their time. The buzzer will ring at 18 minutes. I request the speakers after their talks to be seated on the dais for the question and answer session. I now request the session chairs to lead session 2.
Thank you. I'm going to request Dr. Kaul to chair the session. And just with a very big thank you that you have a pediatric component for anti-tubercular therapy, which is, just so, it's just so painful to see them when you're sitting in the OPD, not getting proper treatment. So, for you and the speakers.
Thank you, Dr. Bhatnagar. I think we have Dr. Sharman Singh. I have known him. My first talk in AIIMS was from an invitation from him. So I don't forget a fantastic congress he organized at that time. He's the ex-director and chief executive officer of All India Institute of Medical Sciences, Bhopal. Has more than 30 years of experience in various faculty positions, including AIIMS Delhi. Currently, he is the professor and mentor of Medical Sciences and Engineering Center at IISER Bhopal. Professor Singh is a fellow of more than 7 national and international academies, including Royal College of Pathologists and Royal Society of Chemistry, and member of more than 20 national and international scientific bodies. He has been awarded several national and international awards, and including the Dr. B.C. Roy Award by President of India. Professor Singh has 7 patents to his credit, has successfully undertaken more than 46 research projects from his various national and international funding agencies. His major research has remained tuberculosis and leishmaniasis. He has written 6 books, 46 book chapters, and more than 355 research papers. He has been on world's top 2% scientists for last 3 consecutive years. Welcome, Professor Sharman Singh.
Thank you, chairs, and thank you indeed, the organizing committee, really to giving this opportunity to share some of my views. As the title indicates, so I will take you, and maybe Dr. Kaul may take some, lead from here to develop something as a pharmaceutical product, because definitely the issue which I am talking about is tuberculosis, but more importantly, one particular strain. Now, this slide, previous speakers, they have already discussed about, so I'm not going to re-emphasize that tuberculosis, how important it is. Obviously, we all know that primary focus is lungs, but it can also, in fact, any other organ, that what we call EPTB or extrapulmonary tuberculosis. Causative agent also we know. Now, coming to the distribution of tuberculosis, despite Dr. Soumya very nicely, she talked about Dr. Anil Kaul, also bedaquiline or many other drugs, they are there, and lot of efforts globally and nationally. Unfortunately, despite all these efforts, the tuberculosis still remains a major problem, which is indicated in this picture, which is taken from WHO. Now, we know that TB is curable, provided we diagnose early. The initiation of treatment is initiated early and correctly. The third component, probably many of the clinical colleagues, they may or may not agree, but for the diagnosticians, it is very important, and I will take you to the next slide, when WHO also re-emphasizes that correct speciation is important. And this is why I am saying, I will take you my talk, that if you are not doing correct speciation, probably your treatment may be misdirected. Another issue, which is a programmatic issue, you have to have regular supply of the medicine. Sometimes we do see in some of the countries, sometimes even in India also, that there is irregular supply. So then there is a gap in the treatment. And on the side of patient also, equally it is important that patients should also comply with that, and that leads to, if they are not complying to, obviously various kinds of relapses, multi-drug resistance and other things. And obviously, if they are not there, then you can have MDR and XDR. Beside these, in the morning, in the keynote address also, Dr. Soumya also highlighted that we know for sure that old is also one of the component which is, drives to you. And then, of course, there is comorbidities and smoking as well. The success rate, this slide shows that despite all efforts, the success rate, you can see that even in 2019, which was analyzed and released by WHO in 2022, here you can see it is less than 60%. So about 40% patients still they are not having success for various reasons, and other things are there. So this is MDR. So unfortunately, no one in this audience will like this kind of picture to see that the bubble is biggest in India. So still we are having the maximum number of cases of MDR in tuberculosis. It is not that government is not trying. Government has done a lot of, and you can see that there is a massive expansion of services for the treatment and diagnosis of tuberculosis in India, including various other medicines. Here you can see that earlier there were very few, only 2 national reference laboratories. Now we have 6 NRLs, or what we call national reference laboratories. We have 34 intermediate reference laboratories. We have 58 where you can do phenotypic culture and sensitivity. We have 3,660 experts or TrueNet. It is a huge number for any country. And then we have, for treatment, more than 2,000 DMCs, or what we say direct microscopy centers. We all know, I will not re-emphasize, GeneXpert has made it became the game changer. It has made a lot of, changes. And another diagnostic and used for the drug susceptibility testing is, because expert detects only rifampicin resistance, and obviously that is used as a surrogate for MDR. Now WHO is also thinking that, whether the definition should be changed or not. So for the actual multi-drug, means INH and rifampicin and other drugs also, that is what we call second line drugs also. This is the line probe assay. But the gold standard still remains the culture. We know that this is the MIGIT one, which is government approved, and it is supposed to do the highest sensitivity in the detection as well as for the susceptibility testing. Now,
why I am re-emphasizing, because if you see even this document of 2022 of WHO, it emphasizes the current guidelines for treatment of drug-resistant TB stresses need of access to reliable, quality assured drug susceptibility. This is very important. And therefore, various national TB control program, they have the certification system also. This should be provided by national TB programs and associated laboratory, means here the private partners also. We know that rapid molecular testing, that means what I mentioned, the expert and LPA, they are there, but it is important they should be utilized properly by all the program managers. It is also important that DST of these drugs is essential to select appropriate treatment. Here, probably Dr. Anil Kaul said, so I will little bit disagree because he is a company person. In the program manner, he said that sensitivity is not required, but MDR is required. So for rider is there, yes. If you are not suspecting MDR, obviously there is no need, even today. But if you are suspecting MDR, the susceptibility has to be there. Now, we know that molecular techniques, they have revolutionized the diagnostics and our understanding also. All the genes they have been identified, I am not going to name all these, including beta-alanine genes. They confer resistance, phenotypic resistance, but many times they do not coordinate, concord with each other. Sometimes you can have phenotypic resistance, patient is not improving, but genotypically, if you see, there is a resistance gene is there, and vice versa also, it happens. So there are other factors also, because no single resistance gene is having 100% resistance phenotypically. Even the best one is rifampicin or RPOB, that is also 95%. Now, the new thing which probably in India is being missed, but now it has been slowly and slowly being realized, that there are many patients where you have heteroresistance. Heteroresistance is where the patient is harboring the drug susceptible as well as drug resistant strain. Both are there. And here, one study, very important study, and that made us understand that if you see here in this slide, you will realize that the gene expert, what we see here, this is gene expert, it can detect resistance only when you are having heteroresistance, means you are having 50% resistant strain, then only it will say resistant. Otherwise, it will give you false positive, means susceptible. Even though patient is having 50% population of mycobacteria in your body, which is resistant, which is usually missed, and what will happen? That the susceptibility testing is not done, and then obviously these patients, they will later on resistant. And so best treatment initiation should be based on the midget, that is liquid culture. Here you can see even the 1% resistant mixture is there, it will detect resistance. So that is the most sensitive. Now this was again done, so I will not go. This heteroresistance I would like to re-emphasize here. So here, those I am sure that everybody does the molecular biology. You are from, most of you are from basic science. So here 2 peaks means this is heteroresistance. So the population is having susceptible as well as resistant molecules. Now we were worried because in last 10 years or so, Northeast has been given a lot of emphasis, lot of money by DST, DBT, and DHR as well. We realized earlier the Northeast people, the investigations were not that much. And last 10 years or so, we realized that many of the TB patients, they are having more resistance, more MDR and XDR as compared to the mainland. And then this was seen in other parts also. And then we saw that the Beijing strain is more common in Northeast. Later on, we found that it has reached up to various other parts of the country. So this is a polygotype. So various lineages of the mycobacteria, they are there, they are identified. So the first one is Beijing strain. So this spacer 1 to 34, they are missed in that. So that is very peculiar. So here is polygotyping. Here you can see that CAS, this is a Central Asian strain. This is of course most common, But second one is Beijing. And this is not very old, about 5-6 years old studies. We saw that Beijing has made inroad in the other parts of the country, including Punjab and of course, other states also. So then we also see here that, drug resistance, if you see in HIV positive and HIV negative, both here, the Beijing was very, very common here almost. Here you can see about 16% cases. We saw that some people, they advocated that probably some of the lineages, they have some organ specificity. We didn't find any, even though there are some reports from other parts of the country. And therefore, from Pan-India, we collected strains from all parts of country and we geno-analyzed. And what we found that obviously the Assam, you can see the maximum is Beijing. You can imagine also because it is near to China. But in other parts also it was there. And if you see in neighboring countries, Myanmar here, that is also having very high concentration or prevalence of Beijing strain. This I will not show again, this is again Beijing, very high percentage. In here, this slide shows that MDR, if the strain is Beijing strain, then you have very high chances of drug resistance. So about, you can see more than 50% of the isolates, if they are Beijing, they are going to become M. The Beijing is also very peculiar that it is having high transmissibility and clustering. We also knew that the other countries and other scientists, they have also reported that this is highly virulent and has good transmissibility and is epidemiologically also, if you see, that the prevalence of Beijing and the drug resistance, they are going hand in hand. But obviously what we were not knowing that in India, whether it is happening correctly or not, and if this is the Beijing only in the Northeast, and if it is related with the resistance, what are the regions, what are the various sequences, and what we can do in the Beijing strain. So what we did, my student Bean is, see, we did 11 genomes of our Indian isolates and 288 genomes, see, downloaded from the NCBI. And so out of 299 genomes, what we found that the, not found, sorry, this is inclusion criteria. So 158 were genomes from African origin and 39 were from Asia, not included 11 of ours, and 99 were from Europe and only one was from America. So here you can see clearly that obviously this is a lineage, Beijing lineage. And what we found that the other conditions, they were almost same, which are reported GC content 65 and the hardcore and the softcore. Both we analyzed and what we found almost similarly, GC contents were also same. The novel coding sequences, what we found that the out of 64 shared accessory genomic elements we found, the genomic elements were involved in the metabolism. So means these genomes, they were involved in the metabolism, cell storage and signaling. And some were also in the information storage processing. But interestingly, we found that coding sequence of 1810 base pair and 592 base pair were found to be absent in all the lineages of genome 1 and also in the other strains to some extent, but not in all. This included part of the gene deletion RB1524, RB1525, and 3651. Now here I would like to emphasize here because we are dealing with the Beijing. So lineage 2, if you see that the CRISPR associated endonuclease-Cas2, that was also deleted. Those who are working on CRISPR, they know more than me that CRISPR is very, very important, and Cas1 and Cas2 proteins, they are very important proteins which derive the metabolism and other reactivities. So here, this was deleted in the Cas2 sequence were deleted in all Beijing strains. So that was very, very important. Here you can see there is no no amplification. This was confirmed by using that whether this sequence is there or not. So using the PCR, we confirm that yes, it is completely deleted, Cas2. So what impact we found that the Cas1 is most conserved protein in the Cas family, which is predominantly in India, we see. In North India, we have Cas predominantly. In South India, we have East African Indian strain. And as I said, Northeast, it is Beijing strain, which is coming up and it is taking over probably in next few years or so. Probably Cas will be number 2 and Beijing may be number 1 because it is highly transmissible strain. So that was there. And we also found that the integration, which is dependent on this Cas1 was compromised. We also know that Cas1 is highly important and it is involved in the DNA repair. So what does it mean? That if it is deleted, means DNA repair is compromised. And this was, we saw in the Beijing strain. Now, this is important to understand that this observation shows us that if the Cas1 deletion is there, then what happens that we have the IS6110 element, which is a multi copies. In EAE, if we see it is having less than 10 copies, in Cas it is having multiple copies. And then of course, so this element is not having flanking regions. So Cas1 and Cas2, they are gone. So they are deleted. The immediate flanking region of this element, they are intact. And that will lead to the resistance. So that was the very important observation. So this mobile element, which is IS6110, usually has Cas1 and Cas2. So these flanks, they are gone, they are deleted. And the immediate flanking region is playing major role in our drug resistance development. So this is the probably very important observation. So I think this observation needs to be, because this was obviously the in silico analysis. Of course, 11 strains, we also included ours, but of course, huge data, 299 genomes my student analyzed. And I look forward that others, they will confirm this finding. And obviously what we found that, the CDs for the 2816C, 2817C, 2818C, and the others, they were absent. And obviously, this is very, very important. And probably this also indicates that the prevalence of the Beijing in Northeast region and Maharashtra also, Bombay. Bombay is next, other than Northeast regions. In Bombay, we have seen maximum strains of the Beijing, and that both the findings, they corroborate, and we can see that this is probably the region that we are having resistance. We can also, anecdotally also, we can see after about 20 years back when Beijing was not that common, from the old data, the resistance rate, MDR rate in India was much, much less. And now after probably, this is the driving force when it is coming to the mainland from the Northeast. Obviously, we are having more and more cases of drug resistant cases. Thank you. So this was done, sorry, just a minute, please. So the work was done by Saeed Binis Rufai and Umay Kulsum, both my students and postdoctoral. Thank you very much.
Thank you. Thank you, Professor Singh. Would you like to come and sit here? Thank you. So we'll hold our questions for the end of, after all the speakers have finished. It's my pleasure to introduce Dr. Subhash Babu, who's a physician scientist, from India, NIH scientist and scientific director at the Indo-US Collaborative NIAID International Center for Excellence in Research Program in Chennai. Dr. Babu is an authority on helminth and TB immunology and has contributed to major achievements in translational research in the field, including seminal and landmark work on the interface of these infections with other co-infections, metabolic diseases, and multimorbidity, including diabetes, tuberculosis, helminth tuberculosis, and helminth diabetic interactions. With over 200 publications in international journals and extensive global research collaborations, Dr. Babu is highly respected in his field and has received several prestigious awards. We are going to hear him talk on identifying host biomarkers to aid in the diagnosis of pediatric tuberculosis. Thank you so much. I'm so glad the pediatric population is being discussed.
Thank you so much for those kind words, and thanks a lot to the organizers for giving me this opportunity to come and talk here. So I'm going to be talking about some of our work on the plasma biomarkers, trying to identify pulmonary tuberculosis in children. I don't think I need to give this introduction to this audience, but just so that everybody is up to speed. Childhood tuberculosis is a huge, huge problem. We know that about 7.5 billion children get infected with TB pretty much every year, out of which about 1.1 million children go on to actually develop active TB. And about 226,000 children die because of TB almost every year. So it is a problem of tremendous magnitude. And the reason why childhood tuberculosis is also of great importance in terms of public health is it is a very, very difficult disease to diagnose. So pediatric tuberculosis, the main problem is children do not expectorate sputum very well. So it's hard to identify sputum smears and cultures and the molecular diagnostics that are typically used for pulmonary TB in adults. Children also have what is commonly known as a paucibacillary form of tuberculosis. So the bacterial loads are very low. So the sensitivity of most of the diagnostic tests that are available are actually very, very poor. So we really need new tools in order to identify and help aid the diagnosis of tuberculosis in children. So one of the ways in which the diagnosis can be aided is by trying to identify either host-based or pathogen-based biomarkers. And that is something that has been extensively explored in the field of tuberculosis over the last few decades. So if you actually look at what the biomarkers do within the spectrum of TB, we know that there are either risk stratification biomarkers or diagnostic biomarkers. So when I talk about risk stratification, it means basically what are the biomarkers that can help identify people who can, who progress from no disease to MTB infection. So this is a risk stratification for infection. You also have people who develop from infection to active disease, and there are risk stratification biomarkers that can help identify them. And more importantly, and as far as my talk here is going to be concerned, it's more going to be focused on diagnostic biomarkers. So there are diagnostic biomarkers that could be useful in aiding infection identification, diagnostic biomarkers for identifying the form of TB that is now being extensively studied called subclinical forms of TB, as well as diagnostic biomarkers for active TB. And of course, there are biomarkers that can follow up and let us know about TB treatment outcomes. But if you actually look at all the WHO endorsed biomarkers that have come out, most of them, as you would expect, are based on pathogen discovery. So GeneXpert, which was endorsed in 2010, lipoarabinamannan, which is again antigen from the bacteria in the urine, was endorsed in 2019. TrueNat, developed by ICMR within India in 2020. And of course, GeneXpert Ultra, as well as C-reactive protein as a triage biomarker for rolling out pulmonary tuberculosis. So if we take a deep dive into what are the different markers that we can try to identify and what are the different approaches that we can use, we know that the pathogen identification is pretty straightforward. You look for either antigen for DNA or other markers that reflect the pathogen load in the host. But you can also look at host biomarkers, which basically are independent of the bacterial loads. And these include antibody responses to your bacteria, host cytokine, chemokine production, metabolites and other proteins, RNA, DNA, as well as transcriptomics, and different mixed signatures. So our approach was basically to focus in on host molecules, including cytokines and chemokines. And I'm just going to give you a brief overview of some of the work that we've been carrying out in trying to identify biomarkers in children. So before we go on, so our study schema is pretty straightforward. So what we typically do, or what we did in this particular study, is to screen children who come to in this case, the Institute of Child Health in Chennai, for symptoms resembling TB, and recruit these children based on different diagnostic criteria, which are either microbiological or clinical or clinico-radiological. So typically TB in children is usually classified into two major subgroups, what we call confirmed TB and what we call unconfirmed or clinical TB. So what I mean by confirmed TB is that these children actually have symptoms and signs of TB, and they have microbiological confirmation of TB. It could be smear positivity, it could be culture, it could be molecular diagnostics. Unconfirmed or clinical TB is actually children who come with symptoms and signs of TB. They have chest X-ray suggestive of TB. Typically, their skin test, TST or IGRA, is positive, and most of it is pretty much all clinically diagnosed. So basically, it's a clinical or radiological diagnosis. They don't have any microbiological diagnosis at all. And the response to TB treatment is basically what then tells you whether these children had TB or not. So it's basically an empirical treatment strategy that they adopt. Unlikely TB children are those that don't have TB, so they don't have chest X-rays that are suggestive of TB. TST could be positive or negative. No previous history of TB, no history of exposure to TB. Of course, microbiology negative for TB. And usually those children who are, have other clinical diagnoses for other respiratory infections, most commonly viral infections, which could mimic cough and fever and other symptoms that children normally present with. So in this particular study, we had 44 children with confirmed, microbiologically confirmed TB. So they were all confirmed, as I said, with a culture, and these are the bacterial burdens. Unconfirmed TB, so nobody was confirmed by culture, so they're all culture negative. And unlikely TB, we had 76. So these are the children who had differential diagnosis that was not conforming to the TB diagnosis. And basically, our study protocol was straightforward. So we collected samples, blood samples at baseline. So this is at the time of diagnosis. And then for the confirmed and unconfirmed TB, because these two groups are, again, as I said, going to be put on treatment. So they get antitubercular treatment for 6 months. So we followed up a subset of these children and collected samples again at the end of the 6 months of treatment from these children. And of course, for the unlikely TB group, we only had baseline samples. And the targeted host biomarker discovery platform, we basically looked at a variety of plasma markers for aiding the host biomarker discovery. So I'm going to go directly into our data. So all my data slides are going to be organized in a very similar fashion. So I'm going to be talking about different parameters. So here I'm looking at type 1 and type 17 cytokines. The y-axis is going to be the concentration of the individual parameter in the circulation plasma. And on the x-axis is going to be the 3 different groups. So you have the confirmed TB, the unconfirmed or clinical TB, and the unlikely TB. And we are basically just looking at statistically significant differences in the levels of these biomarkers in the circulation of these kids. So when we do that, the first thing we found was that we had elevated circulating levels of type 1 and type 17 cytokines, including interferon gamma, TNF-alpha, IL-2, IL-17A, and IL-22 in the confirmed and in the unconfirmed TB compared to the unlikely TB group. So the first thing, the 2 things to note here are the differences are actually between these 2 groups compared to the unlikely TB group, and there were no significant differences between the confirmed and the unconfirmed TB group. When we look at other pro-inflammatory cytokines, we see a similar pattern. So here we're looking at IL-1 alpha, IL-6, and IL-12. And again, all these 3 pro-inflammatory cytokines have a very similar pattern in that they are elevated in the confirmed and unconfirmed TB2 group, significantly elevated compared to the unlikely TB group. So using this set of signatures, we wanted to come up with a parsimonious signature that could completely help us differentiate the confirmed versus the unconfirmed TB group. So we did the ROC analysis using a combination of these markers to come up with the best signature that could discriminate confirmed and unconfirmed TB, or confirmed and unlikely TB. So as you would expect, confirmed TB easily differentiates from unlikely TB with a variety of these combinations. But that's not as important for us because we already know that these children can be diagnosed microbiologically. So you really are not focusing on these children. It is these children that we need to actually focus on because they are not microbiological positive, and it's very, very hard to diagnose them. But what we found is that if you look at a combination of biomarkers, especially a signature that consists of TNF-alpha, IL-2, and IL-17A, we get pretty much 100% specificity and sensitivity with an AUC of 1 in differentiating in this particular cohort of children, the unconfirmed TB group compared to the unlikely TB group. And to add to the evidence of this, we also wanted to ask if these biomarkers are specific for TB and not just non-specific elevation due to inflammation. If you treat TB, then these biomarkers should, the elevated biomarkers should come back down. And that's exactly what happens. So if you look at the subset of children in which we had follow-up samples, so if you look at their blood levels of these elevated cytokines, we see that different gamma, TNF-alpha, pretty much every cytokine in this case that we looked at, all significantly decreased. This is using the confirmed TB group, suggesting that this is basically a host response, probably which is more reflective of the TB bacterial burden, because once these bacteria are gone, so these children at this point are all culture negative, you also see a significant decrease in the cytokines. But more interestingly, the same pattern actually is seen when you look at the unconfirmed TB group. So here in unconfirmed TB, remember there is no the gold standard because they were not culture positive to begin with. So you really don't know what happened except for clinical improvement. Because even if you do a culture here, anyway, they did not have it at the beginning and they're not going to have it now. But what we see, which is very reassuring for us, is that these cytokines that we are using for our biosignatures are actually significantly decreased, even in the unconfirmed TB group. So we also wanted to look at, as I mentioned in the beginning, a variety of other biomarkers, because one of the problems with biomarker studies is that it's very, very hard to validate these biomarkers because you have to be host biomarkers. There's so much variation. There's so much background effects depending on a variety of other co-infections, comorbidities, socioeconomic status, nutritional status, and so on. So the more the biomarkers that we have, we thought it'll be more, it'll give us more strength in trying to validate it in other populations. So we, at least in our small cohort, we wanted to test a variety of biomarkers. So we continued on to look at the chemokines here. And again, we see a very similar pattern with a small subset of chemokines. So in this case, CCL1, CCL3 among the CC group, and CXCL1, CXCL2, and CXCL10 in the CXC family of chemokines that are significantly elevated in the confirmed and the unconfirmed group compared to the unlikely TB group. And again, as I mentioned, so these the combination of 3 chemokines, CCL1, CXCL1, and CXCL10, sorry, actually, of course, with the confirmed group, it's pretty much 100%. But even in the unconfirmed group, we have a very high sensitivity and specificity in demarcating or differentiating between the unconfirmed or the clinical TB children compared to the unlikely TB children. And again, what exactly as we saw with the chemokines, I mean the cytokines, the chemokines that were elevated to begin with are all significantly decreased following 6 months of antituberculosis treatment in these children, both in the confirmed group, which is what I'm showing you here, and in the unconfirmed group as well. So CCL1, CXCL1, and CXCL10 are all significantly, they all significantly go down. The final set of slides, I'm just going to tell you about some other biomarkers that we've also been looking at. So one of the pathogenic markers that reflect lung dysfunction and lung pathology in tuberculosis is matrix metalloproteinases and their tissue inhibitors. So typically these are elevated in the circulation of adult TB patients, and we wanted to ask if they could also be elevated in children with TB, and could that be used for biomarker discovery? And the answer is yes. So we see significantly increased levels of MMP1, MMP2, MMP7, MMP9 in children with the confirmed and unconfirmed or unlikely or clinical TB compared to the unlikely TB group. And similarly, that is also reflected in the elevations of the tissue inhibitors. So TIMP1, TIMP2, TIMP3, and TIMP4 are all significantly increased as well. So here again, if you actually come up with a signature of a combination of MMPs and TIMPs, you can actually pretty much completely differentiate the confirmed TB from controls, as well as from the clinical TB from the controlled children as well. And finally, some recent data looking at other inflammatory markers. We looked at 3 different molecules, chitinase, HO1, and IDO, which we had some inkling that these were markers that typically reflect disease activity in adult TB. And we had actually shown that IDO and chitinase are actually very good markers of unfavorable treatment outcomes in adult TB. So we wanted to ask if they could actually also be used for pediatric TB children as well. And we found that both chitinase and HO1 significantly are increased in children with confirmed or unconfirmed TB compared to the controlled children. And so I'd like to basically conclude saying that we have come up with a cytokine chemokine signature, which very accurately serves as a biomarker for diagnosing pediatric TB, at least differentiating pediatric TB from the controlled children. Similarly, we also have good promising biomarkers in terms of the matrix metalloproteinases and the tissue inhibitors. And finally, other inflammatory enzymatic markers could also serve as good biomarkers for TB diagnosis in children, I think. So with this, I'd like to acknowledge the people who actually did the work. So from my lab, from our colleagues at NERT, who were very helpful, who were instrumental in basically the clinical study, doing the clinical study, as well as our collaborating hospitals and our funding sources. So with that, I'd like to thank you very much, and I'd be happy to answer questions at the end of the session. Thank you.
Thank you, Dr. Babu. Please.
So next online, we have Dr. Vijaya Lakshmi Velluri. Dr. Velluri has worked on various, yeah. Oh yeah, the topic of the. So the topic she will talk today is immunity risk factors and biomarkers for TB in children and adolescents. So a brief summary about herself. She, as I said, that she has worked on the various aspects of immunology and genetics, mostly related to tuberculosis. Her contributions to science include a demonstration of in vitro correlates of immune protection to study the efficacy of BCG vaccination in children and potential use of most immunogenic protein of BCG vaccine, antigen 85A. She was one of the first investigators in India to report the effect of BCG vaccine vanes after 5 to 7 years. She received several projects funded by various government agencies such as DBT, ICMR, DST, and also by NIH USA. She has more than 120 publications to her credit. Welcome, Dr. Velluri, and looking forward to your talk.
Thank you. Yeah. Am I audible? I would like to start with by thanking the organizers for giving me this opportunity. A very well organized conference, I should say. So that's the topic: immunity, risk factors, and biomarkers for TB in children and adolescents. I don't have to stress the importance of TB in children, but yes, out of the 1.3 to 1.4 million deaths every year, about 100,000 of them are children. They die of TB. They could probably be more susceptible because of their immature immune system, and some co-infections like HIV could add to their susceptibility. We all know that it is difficult to diagnose TB in young children. And so it is important to understand the immune responses of TB in these children. This is especially important so that we could plan strategies for prevention of TB, immunotherapy through immunomodulation, or also to develop immunodiagnostic markers. We performed 2 cohort studies, one specifically in children and other in children and young adults. The first one, of course, I did several years ago, but then I would present it because it still holds good. And also it would form a preamble for the second cohort study. This was under the mentorship of Dr. KJR Murthy, who was a clinician, and each time he would see a child with TB in the clinic, he would be upset and he would say, as an immunologist, can you explain why a child who's been vaccinated with BCG gets TB? And so we did plan some cohort studies. We would visit vaccination centers and enroll infants and document every detail, such as vaccine administration, how it would be administered, the type of vaccine that is administered. And to our surprise, we saw that there were several strains of vaccine which were given to these children. There were 3 strains at least. One from the BCG lab itself, Guinea lab. The other one is a Glaxo, and the third one is a Japanese strain. So we did document all these other demographical details, and we would examine the ulceration at 6 weeks and whether this would lead to scar formation, and also follow them up periodically for development of TB. We did carry clinical and immunological investigations in these children, and some children we did follow up for up to 7 years, which was quite tough. I would briefly mention the major observations of this study. To the right, I'm just presenting the ones that are published. So we did say that the tuberculin skin test is not the right indicator to indicate the effect of BCG. In vitro tests are definitely better. And we realized that about 70% of the children who were vaccinated did benefit from the vaccine, from BCG, but with age. So there's some peptides. We did test all the peptides. These peptides we isolated in the lab of Dr. Patrick Brennan in Colorado, because they had the expertise to do this. And we tested them in children by using them for stimulation studies. And we saw that the cluster between the 34 to 30 kDa appeared to be the most immunodominant, which was later also reported by other investigators. So we suggested that the waning immunity could be boosted using this modified R32 kDa, the antigen 85A of BCG. Also, we saw that there was polymorphism in some of the children in the interferon gamma gene, which resulted in low levels of interferon gamma, also scar failure. So are we correct in blaming the vaccine all the time, or is it the child's host immune response? To look at other interferons, we also saw that there was significantly higher expression of IL-15 mRNA in the antigen 85A stimulated cells, which was also a marker of protection. So are we correct in looking at only interferon gamma levels was the question that we asked. So we concluded that the efficacy of BCG, if there is a scar, in which 70% of the children, they would benefit from the vaccine. Those who do not form a scar do not benefit. Of course, those who benefited also, the effect would wane only for 4 to 5 years. And the best strategy would be to give them a booster. But then what is the best vaccine candidate. In children who do not form a scar and have low interferon gamma levels, is it the influence of a genotype of interferon gamma? There could be a mutation in the genotype. And so is it important to screen for genotypes before blaming the vaccine? This already was shown by Dr. Gokhale. We can see that between the 5 to 14 age group and the 15 to 24 age group, there is a sudden spurt in the incidence of TB. So we would recommend a booster probably at this stage between these two groups. So when we're talking of an effective and an efficacious TB vaccine, there is definitely a need for an improved understanding of protective immunity. And we have to define, as we have heard the previous speakers also, and validated correlates of protection. We need to validate immune correlates in field efficacy trials that would ultimately enable the development and licensure of vaccines without the need for expensive or time-consuming field efficacy studies. I'm just quoting one paper that I read recently here, published in 2022. Antigen 85A specific IgG is elevated in infants that were subsequently infected with MTB. So there are some markers which would tell us that these children would not benefit by giving BCG. Infants, of course, they are particularly vulnerable to the TB disease, but adolescents, a sharp increase in TB incidence between the 15 to 25 years of age, emerge as a target age group for new TB vaccine trials. They develop the adult type cavitary TB disease that is more readily transmissible compared with infants. So this adolescent group becomes very important to stop the spread of TB. This has been adapted from Cranmer's publication. So there are different vaccine candidates which are being tested, both in infants as well as adolescents. Some of the ones which are in italics are already in the last phase of the trials. We are hoping that there would be some good results. So I think all of us believe that there are biomarkers to discriminate between protection and inflammation is important, which could facilitate design of both vaccines and drugs. Having said that, I will now come to the second cohort study as part of the Indo-US TB Vaccine Action Program. We are part of the Report India Consortium. This is jointly funded by the Indian government and the US government. And I do collaborate with one, Dr. Ramakrishna Vankailapati, who works at the University of Texas in US. Our work includes identifying immunological markers of persons at highest risk of progression of latent TB infection to active TB. We also worked on innate immune cells, both in adults and in children. And while doing these projects, we also developed or improved the existing method of IGRA. So we have an in-house IGRA for testing the susceptibility of individuals for TB. So for this presentation, I'm going to show the data on immune responses that make children who are at a great risk for progression from latent TB to active TB. So we started with, on day 0, these are household contacts of pulmonary positive patients. We followed them up periodically, once in 4 months till 2 years. Sorry. And we did clinical evaluation as well as immunological studies in them. we saw that young household contacts between the ages of 15 and 35 years developed TB. So out of the 22 who progressed to TB, about 14 or 17 of them were young household contacts. These are the cell subsets that we saw using antibodies, fluorescent tagged, and using a flow cytometry. This is a busy slide, but I think the next few slides are going to be busy. We have several. Could I have some? Wow. Water, please. Sorry. I'm sorry about that. Please get me. Sorry. So these are the T cell subsets that we evaluated. As I said, the slides are busy, but I'm going to summarize them. This is the heat map that we're going to show. These are from different age groups, 15 to 35, 35 and above, and under 15. As you can see, all the cells, there are differences. These are all normal people, and there were differences in the cell types at different age groups. This again is a summary of the previous slide. So as you can see, under 15, it is mostly CD8 and CD4 T cell, which is highly present. In the 15 to 35 age group, it was CD8 cell that was dominant, whereas about 35, the cells were different. So there are different cell types in different age groups that seem to be dominating. So when we plan for a vaccine strategy, I think this needs to be kept in mind. Against the CD4 cells, I'm going to summarize it again. The CD8 cells. So this is a summary. We saw CD4 T cells higher in the LTBI positive, irrespective of the age. We saw the naive CD4 T cells in the LTBI positive, which decreased with age in the LTBI negative. Children who were less than 5 years had more of the CD62L, more of the memory cells, and these cells also decreased with age. So we did see different cell types, as I said, which differed as they grew in age. And the most important point here is we observed switch from CD4 to CD8 T cells from age under 15 to 35 years, and loss of memory markers after 35 years. I'm sorry for that. I have a bad throat. Similarly, for the NK cells, the cell panels were different in different age groups. Again, the major observation here was there was a switch from high T cell subset from age less than 15 years to an increase in the NK cell subset from 15 to 35 years, which continued after 35 years. Similarly, the Treg subsets also differed in different age groups. Monocyte subsets followed the same. They were different in the 3 age groups. Because most of them were young adults, we also tested their serum hormones. And children who were between 15 to 35 years, We did see an increase in T3 hormone, DHEA, vitamin B12 and cortisol, but there was a dip in T4 calcitonin hormones. This is again a heat map showing the plasma metabolites signature in both children and adults. Some of them had HIV, others did not. Again, a summary of the signatures of these children. Between the converters, between active TB progressors, and between the converters in adults. So what I'm trying to say is there are differences in different age groups. So when we do plan a vaccine strategy, we need to look at which cell type is more dominant and which vaccine would again, the metabolite analytes of LTBR resistors in children versus adults. Yeah, I'm about to finish. Thank you. Again, the cell types differ between adults and children. We also compared the antigen 85A versus CFP10 and ESAT6 specific interferon gamma levels across different age groups, and we found them to be different. To summarize this, it was interesting to see that in the non-converters, when they were stimulated by C+E, that is ESAT6 and CFP10, the levels were low, they differed. Well, with antigen 85A, it was high. In the converters, it was reverse, whereas in the progressors, they were both high. So we could probably use this as an algorithm to see who are going to progress to active TB. So we were wondering whether there were markers of protection versus inflammation. So I'd like to summarize by saying that there is a switch from CD4 to CD8 T cell subsets after 15 years. There is a loss of memory phenotype after 35 years. There is a dominance of natural killer cell subsets after 15 years. The cytokines also differed between ages, between these age groups. And hormones, of course, in the young adults, that is between 15 to 35 years, they did have high T3 hormone, DHEA, vitamin B12, but low T4. So I'd like to go to the translation implications of all this. So when we contemplate on a booster vaccine at about 7 years of age, which is important, we should include different antigens in tests and test both the protective versus the inflammatory responses. This is useful for assessing new vaccine candidates. A screening of adolescents, that is young adults at 15 to 30 years who are at a high risk for developing TB, and also screening for serum hormones, T4 and calcitonin is important. Treatment of the target population, that is young adults, seems to be effective to prevent the spread of tuberculosis. I would like to thank my group, both at BMMRC and at the University of Texas. Thank you.
Thank you. Please come and take a seat here. Thank you. Again, it's my pleasure to introduce Dr. Amit Mishra. His research, so Dr. Mishra will be talking on orally inhaled dry powders for treatment of TB, Sentinel, second-line drugs, and host-directed therapies. His research interests include pulmonary drug delivery in tuberculosis, with special reference to host pathogen dialectics, among other critical disease domains. Dr. Mishra has received appreciation as an active research collaborator with several academic and industry-based research groups in India and a few abroad. He's published 70 papers as first and/or corresponding author, apart from 15 popular science, sociology, economics, general articles, and has contributed to 10 book chapters. So welcome, Dr. Mishra.
Thank you for the kind introduction, Shinjini, and apologies for throwing a tantrum. But I threw a tantrum because I'm going to talk about my work as a pharmacist, as a compounder. And I believe over the past 10 years, we compounders have been unnecessarily diffident, especially since Professor Bishai in the audience raised the question about inhaled amikacin for NTM lung disease. So for the past 25 years, our group has been working mainly on tuberculosis and other things that I will talk about later. And it's really fortuitous that we recall that Koch's discovery was published in Awadh Panch in the same year that it was announced in Berlin. And the English translation is nothing if you actually appreciate the Persian part. They call Dr. Koch, जर्मन के मशहूर तबीब ने अपनी इल्मी तहकीकात से तपेदिक का मुजर्रब इलाज दरियाफ्त किया है. And I bring you greetings on the anniversary of this date with the admonition that the two ladies supporting the frame are Hikmat, that is medical care, nursing, bedside care, diagnosis, and Tibbiyat, which is pharmaceuticals. And therefore, I bring this presentation to you to wake you up just before you admit that, oh, we missed something. Yes. So we've been trying to involve really big doctors, key opinion leaders. We've enticed Diacon to Groningen to help us design a trial. We got Helen McShane to Parma to speak to us about the inhaled vaccine. She did go into vaccination, but she used nebulization again, which, well, might have contributed. So I'll talk about CDI formulations. We have several of them. We have first-line drugs, second-line drugs, host-directed pharmacotherapy, and host-directed biologics, all to be given as dry powders for inhalation and with payloads of isoniazid and rifabutin, which is Sentinel, our flagship. Second-line drugs, we've done a combination of D-cycloserine and ethanamide, which are almost never prescribed together. And we've shown how pulmonary delivery and the effective first bypass of the liver enables you to have therapeutic concentrations for much longer if you have inhalations. We have done clofazimine with our collaborator Bernard Fourie in Pretoria. We've done sutazolid, TB Alliance compound 354, which had neurotoxicity, reversible, but then it was taken off because of the toxicity considerations. So we did it at targeting. With Auckland University compound 32625. Host-directed pharmacotherapy has something like nitric oxide donors that do not need a complicated mechanism, but just generate nitric oxide inside the cell, which is nitric oxide, isosorbide mononitrate, beta-NO. To induce autophagy, we use the canonical rapamycin, sirolimus, to explore whether host-directed therapy by inducing autophagy by either sirolimus or nitazoxanide, which has some minor activity against microbial tuberculosis as well. And biologics, we've done gamma interferon DNA for transient transfection of the epithelial surface of the respiratory tract and the lungs, and which has given rise to a very interesting observation that you may upregulate the cytokine, but at the same time you see suppressors of cytokine signaling, SOX and cis-H genes kicking in. So we are trying to knock these down using morpholino oligos or cymorphs. And the USP in terms of our translation institution is that our first line isoniazid and rifabutin target the alveolar macrophages with an intracellular bolus and also diffuse out from these cells into the lung lumen for a very long time. And from the paths that the drug follows for disposition is not from the blood into the granuloma, but from the macrophages that are being chemo-attracted towards the granuloma, and I'll talk about that later. And we have seen several indicators of the fact that macrophages that have ingested Mycobacterium tuberculosis, unfortunately only a laboratory strain, S37RV, may be rescued from M2 activation by the simple act of phagocytosis of a dirty grape particle. The second line drugs, where I mentioned about overcoming the first pass and of overcoming a pharmacokinetic interaction. With host-directed therapies, and this question came up in the morning as well, since 2011 or so, our experience in preclinical models has always been that host-directed therapy is excellent for healing the host, but it is suboptimal for reducing the bacterial burden, and you need a combination of host-directed therapies as well as aggressive bactericidal agents in order to achieve any kind of benefit that you hope to derive from a host-directed therapy. Our work on translation on biologics is to translate to enhance affordability and access. Instead of gamma interferon, protein actimune, we can offer a non-sterile, handheld, self-administered dry powder inhalation that will give you intermittent or transient gamma interferon in the lung lumen and consequences that follow. So, since I am an interloper here, I must say that we have been funded as of February 2023 for a hybrid phase 1b/2a trial of Sentinel, isoniazid distributin. The others are we have preclinical proof of concept, but no funding to and no competence to design trials to take this forward. Since I see very few pharmacists in the audience, I'll take some time to talk about dry powder inhalations, DPIs, the devices, and the doses. So dry powder inhalations are not nebulization. They are not things that you put into your mouth and press to get a puff and inhale with eye-hand coordination. They are not actually like substance abuse of very, very large doses through the nostrils. They are the simple rotahilar, which very few of the physicians in this audience probably prescribe in India. These are taken in as a dry powder by putting it in your mouth and just drawing a breath. So these make you cough the least. They enable you to adjust the rate at which you're depositing it. And they are, as I said, handheld, non-invasive, non-sterile, outpatient devices that are dirt cheap. So our powder of Sentinel was tested with some people who are developing really high dose inhalers. This is our colleagues Anhaya de Boer and his group at Groningen. And this is the Cyclops. You will notice that this is discolored, which means that our powder is not married to this device. Instead, it is married to the rotahilar, where it is completely taken in. A single dose from a capsule is all taken in. Whereas the material construction of this thing makes it stick to the Twincer device. This is a Twincer device from a company called Pure IMS that Anand de Boer and Eric Freiling have formed at Groningen. And its successor is the Cyclops. And this can really give you high doses. I'll show you just how. I have different kinds of inhalation devices in my pocket, so please bear with me. This is what it looks like. You put it in your mouth and you suck it. You hold your breath, and if all of it has not been taken out, this is transparent, you can see it, you put it back again and inhale. The one thing that you should not do is to breathe back into this thing. This is a bit of a patient education glitch that this high dose inhaler is talking about. But for our purposes, for the Sentinel trial, we are happy to use the rotahilar and developed by Cipla. Doses wise, we know that inhaled insulin went to the market. The dose was 0.4 mg. Inhalation time was about 3 seconds. 3 seconds. Tobramycin, which is given for opportunistic infections and CF, is given at 250 milligrams. Amikacin I have not listed here because it's given by nebulization. Capromycin has completed a phase 1 trial funded by the philanthro-capitalist organization called MEND, but they withdrew funding after the phase 1 because the objective of the foundation was to make a significant impact on the field, and the significant impact was made. So Tony Hickey's group, who's done this formulation and done the trial in South Africa, is sitting there. But the problem with this is that they've given 300 milligrams by inhalation, However, their technology does not allow them to have a single dose. So they have patients inhaling 12 capsules, one after another, to get a total dose of 300 milligrams and 10 seconds for each. So that's a 2-minute period for which you have to inhale, which is not convenient for the patient. Just out of interest, the highest dose that I could find in the literature that has been given as a dry powder inhalation is in the bad old days when doctors wanted to see how a patient would tolerate common salt, is 635 milligrams in 10 seconds, and FEV, lung function, everything returned to normal baseline levels within a period of 10 minutes after inhalation. Nebulizers are suboptimal for deep lung delivery, and we've known that since 1974, where it was shown that 1.3 to 13.7% of the dose was deposited in the lung, and within 1 hour, anything deposited in the airways, as a nebulizer does, and as it should, because it is designed to work for asthma, which is bronchoconstriction, not for pulmonary delivery. Anything deposited in the upper airways just disappears only within 1 hour. So this has been known since 1974, but also recently confirmed. Smaldon, Gerald Smaldon and Samuel, they published in 2020 that with the most advanced technology of jet nebulizers, you do not get above 25% deep lung deposition if you nebulize. You need a dry powder inhalation. Okay. The other problem in developing Sentinel as a dry powder inhalation for pulmonary delivery in tuberculosis is that regulatory agencies are informed by the pollution people. They talk about deposition in the airways and the lungs in terms of suspended particulate matter, which involves exposing animals to 5 hours or more of exposure, to not only exposure, to a very dilute aerosol. This is in stark contrast with what we have, which is a bolus going in a very short time. The entire set of responses by the animal receiving a dilute aerosol over 5 hours to 6 hours to 8 hours, and an animal receiving a powder in a very short span of time are quite different, of course. In preclinical research, the field was dominated by an apparatus called the Penn Century apparatus, which I wonder if many of you might have seen. It means it's a laryngoscope for rodents. You hang the rat by its teeth on here, use a laryngoscope to intubate, and you use positive pressure in a syringe to intubate and push down the powder into the respiratory tract, which of course is very, very different from ambient pressure inhalation of material. So we invented an apparatus that comprised of a 15 ml centrifuge tube. We made one hole in the side where the animal's nares could be inserted. And please remember that rodents are obligate nose breathers. So although I said that we are not snorting cocaine, my rodents are all inhaling from the nose. There's another hole in the top in which you introduce a tube and a rubber bulb. So when you press the tube, you aerosolize bright turbulent air, and the atmosphere, the animal is breathing in a very dusty atmosphere. And you control the dose by controlling the time for which you hold the mouse against the apparatus. The only thing you have to remember is you have to press the rubber bulb and not the mouse. Okay, that was well flat anyway. So we use this thing for delivering to mice, to rats, to guinea pigs, and if we interface it with a baby mask that Cipla also makes, we can use it for monkeys. And we did our 90 days repeat dose inhalation toxicity studies here using these monkeys. One more last thing I would like to say is that regulators worldwide now want us to give safety data on dogs. And we do not want to do this on dogs for very sound reasons. This is our human respiratory tract and lungs. And this is the cast for a dog. So in anatomical terms, this is monopodial and fractally branching instead of binary branching in the human. And for comparison here, this is a mouse, this is a rat, and this is a non-human primate baby. So number of airway generations, the pattern of branching gives you an entirely different airflow pattern. So when you're doing a non-rodent study for regulatory purposes, please do not use dogs for deep lung delivery. The results of our preclinical development were published in 2021, and people interested may like to go through it. But the title, I believe, is quite succinct. We offer efficacious, safe, relapse-free cure of experimental animal tuberculosis in 1 month if we use Sentinel as an adjunct to human equivalent doses of DOTS drugs. Human equivalent doses of DOTS drugs do not provide complete sterilization of the lungs and the spleen in 1 month. So there are common queries. Why are we using rifabutin and not rifampicin? We would have loved to use rifampicin, except that there's a chemical incompatibility, and this adduct is formed if you incubate isoniazid and rifampicin together. This is also an off-patent compound patented in the 1970s, but it has lower activity than either isoniazid and or rifampicin. But the worst thing is that this smear is unquantifiable and batch-to-batch variable depending on the degree of protonation that you achieve in each situation. So our first publication on this thing says that we wanted to use rifampicin, and we published rifampicin, but this is never going to make a pharmaceutical product because you can't have an active agent in unknown quantities in different batches of the material. So we were working with a company called Lupin at that time, and they recently bought the patent for rifabutin, and they were very happy when we said, okay, rifabutin does not give this reaction, even if you reflux it in 0.1 normal HCl for 1 hour, you don't get this kind of reaction. So the question they asked next was, if they have incompatibility, why don't you make 2 sets of particles? Make one of rifampicin, one of isoniazid, mix them together in the same proportion that you'd like to have them. So this means that you go to a kindergarten class and you have 2 kinds of toffees, one red and one green, and you expect macrophages to queue up like good little kindergartners and take one each of one red toffee and one green toffee. But that is not happening because macrophage uptake is stochastic. And therefore, if one guy runs off with 2 green and 1 red, or one guy takes only red toffees, what you have is a nightmare situation of intracellular concentrations of one or the other agent in different proportions, selecting one or the other bacterium for resistance or not. So this is something we wanted to avoid. Thank you. So we set everything together in the same particle so that it is taken so at least the proportions of the concentrations in each intracellular compartment will be similar. So we are going for an academic clinical trial, and we have gone in for some kind of flag for that as well. In the sense that why are we going to for an academic clinical trial, which is a new term for initiated clinical trials in India, where the results of this trial cannot be submitted to the regulator for seeking marketing permission. So that's fair. I mean, we are going to make this at pilot scale. We are going to make it at CDRI because Lupin just walked out of this project because they said that, well, forget what Lupin said, but essentially it's going to be an academic clinical trial with the clinical site at King George's Medical University. The pharmacy will be at CDRI. But that doesn't mean that it will lack any kind of rigor that is expected of a clinical trial done by a pharmaceutical company. We still have to fill the dreaded Form 29. We have to put in a Form CT10 and Form CT4A. And we are under scrutiny by the IEC, which has been authorized by the Drug Controller General of India to act on its behalf and to approach the DCGI anytime they have any doubts about it. So rapidly now, treatment-naive patients, if they give informed consent, we meet the screening criteria. It's a 3+3 double-blind, placebo-controlled cohort expansion study from 15 milligrams of Sentinel to 240 milligrams for the 3 objectives. First, to find the maximum tolerated dose between 15 and 240 milligrams per day for 28 days. Next, to look at pharmacokinetics, not only the blood plasma, but also in the sputum. And this is going to be somewhat daunting because our study personnel will have to timestamp each sputum sample as it is produced. And we have to determine, we have to develop an assay in LC-MS to find the concentration.
It is the 18-minute.
Yeah, 18 minutes. I'm going to just 2 more minutes. So this is our WHO GMP compliant manufacturing facility at CDRI. This is our Form 37 testing license. Our costing is important, so I'll end with that. 120 mg batch size of 10,000 capsules that I'm going to make is going to cost 8,250, so that the cost per dose is 15 rupees 41 paisa. If we add transportation cost by taxi, AC taxi for 1,000 kilometers, and add DOTS implementation costs, the cost to NTEP at point of care would be about 30 rupees, 27.79 rupees. But that's not all. If we consider the direct cost of NTEP, as the Sinha et al. Paper referred to earlier, or the Floyd paper where Matthew Zignol, a person I respect, is also an author. We project, if we can reduce the duration to 2 months, we save half the cost of DOTS implementation. And rushing through the second line, we have everything published. So this is Gareth, who showed that what we are speculating about macrophages traveling to the granuloma is actually true in the Mycobacterium marinum model of zebrafish. Approved drugs being repurposed, we have nitrooxide, rapamycin, so on and so forth. But if you have the time, if I have the time, can you just look at the number of alveoli and the alveolar surface area that is increased if you have rapamycin along with isoniazid and rifabutin in this group as compared to isoniazid and rifabutin alone. So what we're saying is that we can heal the guinea pig's lungs after it has undergone trauma. This is a recent thing where we talked about transient gene therapy. And one more thing, last slide I can show you, is that the caveats are there. It is not as if this is a foolproof method. This mouse received the powder only in one lung. If this happens in real life, you're not thinking of a single dose, let us say, intervention like a vaccine. You can only think of this as missing a dose in a long regimen. The PK in the infected versus this is not important. The lungs are okay. So end of slideshow. Thank you so much for your patience.
Thank you. Thank you very much. Amit, Amit, please join us here.
So the last guy who will be speaking, okay, sorry. The last lady who will be speaking is Dr. Tamseen Edis. She is from Illumina EMEA, and the title of her talk is Role of NGS to Gain Insights in MTB Biology, Epidemiology, and Resistance Profiling. Tamseen has more than 20 years of experience applying genomics to better understand human health in both academic and corporate setting. As a market development manager responsible for emerging applications, Dr. Tamseen spearheaded Illumina's regional response to COVID-19 pandemic, where she developed a regional strategy to support our infectious diseases customers while supporting key partnership initiative. Tamseen currently manages Illumina's regional marketing development activities for infectious diseases and human genetic disease. Through her tenure at Illumina, she has held roles in commercial and technical departments. So Dr. Tamseen, welcome, and looking forward to hear your talk.
Thank you very much. I'm very grateful to be here. So thank you to the organizers for giving me the opportunity to speak here today. And also am incredibly humbled to be amongst such amazing presenters. So thank you so much for allowing someone from a corporation, an industrial partner, to speak at this conference. I'm really very, very grateful. As you can tell, probably from my accent, I'm not local. So I ask that you bear with me over the next 15 minutes or so. I promise to be quick, as I understand I'm the only obstacle between you and dinner. And I promise not to mention the cricket either. Okay, so I work for Illumina, and Illumina is a manufacturer and innovator of systems that enable DNA sequencing and high throughput genotyping. And because our underlying analyte is DNA, our technology can really be applied in a number of different ways. That can be in research or in clinical settings, and in life sciences, in oncology, reproductive health, and agriculture, as well as infectious disease. During the COVID-19 pandemic, we really saw an increase in the adoption of next generation sequencing in the infectious disease market segment. In fact, we actually think that the COVID pandemic really accelerated the use of this technology by roughly 5 to 10 years. So we've really seen an increased adoption of our technology since the COVID pandemic began, and we're seeing that continue as the pandemic begins to wane as well. So just to mention that we really are driven by a conviction of making genomics accessible to many, not just few. And that's why I'm here today. I'm really, really passionate to hear about all of your stories in the field of tuberculosis disease management and what we can do as a US company to really help enable equity of access and enable reducing implementation barriers to really enable the use of next generation sequencing in tuberculosis disease management in the future. So Illumina has quite a long history in the terms of space of next generation sequencing. We have placed more than 20,000 sequences all around the world, and that's a couple of ours just shown here on this slide. And in the past 5 years, we've placed over 160 systems across 23 Indian states. So we have quite a strong presence within the Indian market and quite a really great channel partner, distributor, Primus, and thank you for all their hospitality while I've been here, who have done a great job distributing our technologies across the country. I've got a number of background slides on tuberculosis, and I think that you are all much more experts on this than I will. So I'm just going to briefly flick through these in order to get to the actual meat of the presentation. We all know there's a huge burden of TB within India, with many cases identified, many cases of drug resistance identified, and a high proportion of extrapulmonary cases of TB identified also. We also know that it's a big burden to tackle drug resistance TB, and it's actually occurred by the actual bacterium acquiring mutations over time that can enable the bacteria to counteract the effects of a given drug. And we've already had some magnificent examples of that from Professor Singh earlier along in the day. Additionally, drug-resistant TB has been shown to spread between individuals. We know that drug-resistant TB is difficult to diagnose and treat, causes more deadly infections, and ultimately is much more expensive to treat as well. And this is for you, Ankit, my Illumina colleague, but he did ask that, and we do stress that we understand outside India the importance of tackling TB in a time-sensitive manner. And I can assure you from someone who's come from outside India that we are doing all we can to, as I mentioned, enable access, reduce barriers to implementation, and help with educational burdens as well to really help enable this technology to be adopted more readily. So the current situation of TB, we know it's a slow-growing bacterium that delays identification of drug resistance profiles. We know that traditional cultures can take up to 8 weeks to provide informative results, but we also know that there are new molecular methods that enable far quicker access to results, but their actual utility is limited. We know that the burden of multi-drug resistance TB is increasing, and sometimes there is inappropriate or incorrect use of antimicrobial drugs. And we know that current molecular tests are target specific. They assume a knowledge of an existing known mutation in the TB genome, and they don't have a priori knowledge of existing technologies. So we know this is the status quo for now, but looking to the future and how we can help was what if we could help enable pathogen identification to a broad range of susceptibility to antibiotics in a much faster time frame. What if we could reduce that to less than 48 hours? And what if we could combine multiple tests into one particular assay? And that's what we at Illumina are working towards in our solutions for enabling NGS-based molecular profiling of drug resistance susceptibility in tuberculosis. So you may have seen on my first slide, it's the 25th anniversary of Illumina this year, and it's also the 25th anniversary of the completion of the first genome of Mycobacterium tuberculosis. So this was sequenced in 1998 and is roughly 4.5 megs. Now, if any of you have a history in genomics or history in creating sequences, doing this in 1998 was a huge, huge challenge. A lot of manual effort goes into creating genomic sequences and in actually annotating the genic structure of those genomes. All this work that can take multiple years can now be done in less than a couple of days. But importantly, genomics has underpinned our understanding of both the biology and the development of assays for TB management. So we now have a greater understanding of the biology, the evolution, and how the actual bacterium transmits from one individual to another. We know that genomes can be used to develop novel therapies, develop new vaccines, and importantly, develop assays for drug resistance detection as well. So now to the meat of the sandwich, this is where we can actually talk about how next generation sequencing can actually really enable greater insights through comprehensive analysis of genomic sequencing. So Dr. Camila mentioned earlier today, there are two main applications of NGS, one being whole genome sequencing and the other being targeted at genome sequencing. So pretty much they do as they say on the tin. Whole genome sequencing, we're generating a sequence, a comprehensive sequence of the entire genome of the Mycobacterium tuberculosis sample. And it gives us output data of a full panel of known and unknown mutations within that particular sample. And it gives us the option for retrospective analysis for newly discovered mutations. As discussed earlier, it gives us the opportunity to identify mixed microbial infections. That's where one individual can be infected with different strains or different bacterial species, and it's recommended by the WHO for drug resistance surveillance. However, we know our technologies aren't perfect, and we know we've got some way to go, that we are still reliant on quite early bacterial cultures for whole genome sequencing to be completed, and it requires quite highly trained personnel and infrastructure. And we also know that there's quite extensive informatic capabilities that are required as well to conduct this technology. Targeted NGS, on the other hand, is a sequencing of a selected region of any pathogen genome sequence. This could be done by amplicon or via enrichment. It's a workflow that can be done directly from specimens, which obviates the need for culture, and it gives us a faster turnaround in WGS. It gives us more comprehensive drug susceptibility determination than molecular DST, and it gives us the ability to differentiate between non-coding and coding mutations. Targeted NGS is also recommended by the WHO for drug resistance surveillance, and it has the same caveats or similar caveats to whole genome sequencing. And this is just how we see workflows that integrate NGS into their standard routine use. Detection via sputum microscopy, followed by PCR-based detection via the expert or TrueNat panel. When WGS is being used, it will require culture, a pre-culture, to generate sufficient material to enable sequencing of the entire genome sequence. But that gives us great outcomes of genotyping, epidemiological clustering, and drug-resistant predictions of both known and unknown mutations. And that gives us a really high resolution for epidemiological investigations. Just slightly differently, in targeted NGS, we begin with the same process, but we no longer have the need for pre-culture to generate sufficient material, and we can get similar results of drug resistance and detection of known mutations and initial information about the epidemiology of those particular samples, which can help resolve heteroresistance and also other strain information. So we have a really nice example how a WGS has identified missed resistance that's been missed by rapid culture and LPA. And this is from a Spanish individual who presented with minimal symptoms of drug resistance, and it wasn't really detected using standard techniques. So the patient was the patient's infected lasted from the first diagnosis from 2019 to relapse in 2013. And WGS was completed on one of our systems and showed that the tuberculosis strain had indeed evolved in that individual over time. So this is some of the resolution that you can get when using next generation sequencing. And many of these common mutations were identified using the whole genome sequencing were drug resistance conferring mutations and were more likely common in high burden countries. We also have additional publications comparing the targeted methodology to PCR methods to identify drug resistant strains. We know that the DeepLex technology, when coupled with PCR or nucleic acid amplification tests, can really provide detailed drug resistance data, and it's much faster than traditional cultured methods as well. This is what I've really come for here today, is that we've identified at Illumina a need to enable market access for a holistic sample to solution answer for drug resistance determination in tuberculosis. And what we've done is we've partnered with who we believe to be a leader in their field, GeneScreen, to help eliminate tuberculosis by 2035. So now customers across the globe can purchase a sample to solution answer for targeted NGS using Illumina technology and GeneScreen technology from Illumina. And we really hope that this will help enable access to the technology across the globe, not just in India. Bear with me for a moment. So GeneScreen is a small company based in France. And they have a small number of employees, but they have an active representation across 80 different countries. And they've produced what's called the Dplex MIC-TB Target Amplicon Kit, which I'm sure you've heard of. This has been mentioned several times today. And this kit's used for TB identification and drug resistance. And what was particularly appealing to us is it has a very attractive software analysis suite. They have an RUO version, which Illumina will be co-selling, and they also have a CE-IVD solution as well. They have proven on-market performance, and importantly, they are an integral member of the Seek and Treat trial, which has been funded by Unitaid to validate the performance of NGS as a diagnostic test for tuberculosis. So this really gives Illumina a full range of opportunities to conduct drug resistance, drug susceptibility determination in tuberculosis. We have an option which requires a targeted solution, which is the Illumina and GeneScreen Dplex MIC-TB combo kit, all of which the features can be found on the left-hand side. So it's culture-free, fast, and can enable the identification of more than 100 species and extensive anti-TB resistance across 15 drugs. We've also got the features of whole genome sequencing and another Illumina product, which is a broad-based culture-free panel for detection of 208. Do you want me to give me 2 minutes? Okay, thank you. A broad-based detection of 280 pathogens, including 187 bacteria, 53 fungi, and 43 viruses, all from the same sample. Just do this and then I'll skip to the end. So the Illumina GeneScreen Dplex MIC-TB combo kit has a number of different features. As we mentioned, it enables prediction of resistance or susceptibility to 15 anti-TB drugs in a very short time frame of less than 48 hours. It can detect heteroresistance down to 1 to 3%. Its starting material can be either culture-free or cultured isolates, and it can be used for MTBC strain typing as well, to aid the identification of multiple non-tuberculosis mycobacteria strains. And as I mentioned, the data analysis is a really secure and easy to interpret option. So I'm just going to skip through all of these. If anybody wants any of this information, more than happy to talk to you about it on another occasion. And I'll just finish up here. We acknowledge that it's World TB Day tomorrow, and happy World TB Day everyone for tomorrow. For those of you who are early risers, in the morning, Illumina will be hosting a webinar series with the company called LabRoots. And if anybody wishes to attend this, we have the QR code shown here, where we have 3 speakers from across the globe presenting on different perspectives of the use of next generation sequencing in tuberculosis management. So we have Tim Rodwell, who's been involved in the FIND-Dx Seek and Treat trial. We have Dr. Melanie Grobelaar, who's from Stellenbosch University in South Africa, and she's talking about implementation of workflows to enable high throughput use of NGS for TB drug resistance determination. And finally, we have Dr. Vitaly Stinchenko from Pathology West at Westmead Hospital in Australia, and he'll be talking about the use of whole genome sequencing for understanding transmission patterns of tuberculosis. So if anybody's feeling like an early riser tomorrow, we encourage you to enjoy that, and thank you for your attention.
Thank you very much. Please join us. We have about 20 to 25 minutes for questions. I'm sure everyone is hungry and it's the end of the day. So does anyone have energy to ask questions? Yes, sir.
This pertains to the talk by both Dr. Vijay Lakshmi and Dr. Babu. I think the difference between confirmed and unconfirmed, one of the major cytokines seems to be Th17. And the same way between the scar, for the childhood BCG vaccination or in infants, do you think that it is the Th17 activation which differentiates between the two types?
I'm sorry, I didn't catch the last part of your question.
Okay, the question is between the confirmed and unconfirmed tuberculosis cases that you described with the cytokines profile, do you think Th17 is a good marker for that? Among all of them, I found that that was the most strikingly different between the two.
Yeah, that's a very good question. So one of the issues with any of these biomarker studies, especially the non-specific host dependent biomarkers, is the non-specificity of the response. And what we've done is we've tried to validate our study using a different cohort of pediatric TB, and we were very successful at validating our cytokine signatures, but we've not yet started doing our validation studies for the other signatures that we discovered. Having said that, again, both the cohorts that we did were all South Indian children, only restricted to a certain geographical area. So our hope is that this needs to be validated by different groups across different areas, as well as different, not just in India, but across different ethnicities, because there is going to be a lot of variation. Mainly because the pathogenesis of TB that is driven in South India is not necessarily the same sort of an inflammatory response that you see, say, for example, in HIV-infected children in South Africa who also have TB, or more Brazilian cohort, which may have a different set of signatures.
The same way about the scarring, Dr. Vijay Lakshmi, do you think Th17 activation is limiting between the two groups?
Yes, sir. Unfortunately, we did that several years ago, as I said.
At that time,
we didn't really look at IL-17 as far as scar is concerned. But then IL-17, yes, seems to be a protective marker. And like you said, it is different. When individuals are likely or who are more susceptible, it is low. And those are protected from TB, it is high. Yes, it can be it is differential.
One question I have to Amit. Amit, can I ask? Okay, Amit, when you talked about that macrophage are taking the particles, originally it was shown in the case of amphotericin B, that amphotericin actually goes first to macrophage and then they go to the site of fungal infection. Same was shown in the case of HIV infection. It's picked up by dendritic cells and they actually then take it to macrophage.
It's a slight difference here,
Yeah.
in the sense that amphotericin B for leishmaniasis or for disseminated fungal infection does not have associated, these diseases do not have associated with them a specialized structure such as a granuloma. In our case, it is very important for us to demonstrate that what we're claiming is that minuscule doses actually target the granuloma and that we have macrophages carrying, taking these as Trojan horses. It is not simply a question of disseminated macrophages taking it.
Are the macrophages or dendritic cells in the same context?
Does it matter? Does it matter? Insofar as what they're doing is that they're carrying drugs. And DC as by definition, DC or even dendritic epidermal T cells, DETC, are minuscule in terms of in comparison. And macrophages, by the way, alveolar macrophages are barely 2% of the population of the lung parenchyma. So it doesn't matter. I mean, if DCs are taking them up, they're welcome to it.
Maybe Professor Tyagi has a question, yeah.
Amit, this question is for you. Granted that the primary location for mycobacteria is lung, but it is all over. For example, in spleen, it hides in.
I'll anticipate the question, sir. That is why you are saying that it's going to be an adjunct.
Question
It's an
first.
adjunct therapy, sir. DOTS is not going to stop. It is over and above the standard oral treatment.
Hear the question, please, first. Yes, Dr. Tyagi, please go ahead.
What happened?
So you said that the tuberculosis is all over.
Yeah, I said it is all over. For example, in bone marrow, in brain, in TB meningitis. So does your method targets bacteria all over in the body?
No, sir. It is meant as.
Where does it do then?
It is meant as an adjunct therapy to shorten the duration of treatment. You continue the treatment, the extended phase.
Only pulmonary.
Sir, you continue the extended phase for 4 months because you want the pharmacokinetics of the drugs to build up in the lesions in the lungs. Otherwise, miliary TB is gone. There's no bacteria in the spleen. By the end of that, at the time of sputum conversion, there are actually no live bacteria in the accessible sputum. It is in the cavitary lesion, in the caseous region, that you probably have surviving bacteria. And you are trying to, current medical practice is trying to douse a fire in a small corner of the room by flooding the entire city. What we are just saying is that if the focus is to have large concentrations, high concentrations in the lungs for a very long time, you need to have a sustained delayed release targeted drug delivery system that will give you the adequate amounts at much less input. So that targeted drug delivery as
Excuse me,
highly.
could we keep the questions and answers brief,
Sure.
please? The others also need to speak. Anyone, others?
So I have a question for Dr. Babu. Subhash, there were several metabolites that you studied where there was significance in adults that you've shown before, but not in the pediatric cohort, for example, IDO. And why do you think that is the case? And is it something to do with the differential T cell response that children have?
So I'm not sure exactly why we didn't see any differences in IDO. So that was your question, right? So yeah. So what we did find and published earlier is that IDO is actually a very, very good marker for recurrence in tuberculosis. And so if you individuals who have differential levels of IDO actually are much more prone to have TB recurrence, but this was in adults. In children, what happens, what we see is that because IDO probably is, comes up with both respiratory viral infections as well as respiratory bacterial infections, we are not seeing this difference. But what we are now trying to find out is, is it a good biomarker for, not for diagnosis, but for prognosis, meaning if you follow them up, will there be differences once you treat the bacteria?
I was just wondering, following up your answer, Dr. Vijaya showed in her presentation a very nice, it was a very stark metabolite comparison between children. I had never seen such a comparison, so black and white kind of a thing. So how does that correlate with your findings? Did you look at IDO in your metabolite panel, Dr. Vijaya? It's a question for both of you, basically. Yeah.
Yeah, we did find it. Even we were surprised to see such a stark difference, as you said, between different age groups and how there's a switch between the types of cells. Yes.
And how many metabolites did you really pick up?
Plasma, of course, there was an area of metabolites that we did, but then we couldn't present all of them. We just presented the top ones. So I'm not sure. Do you remember the number? Yeah, we did a huge.
Take the mic, please.
It's about 330, and then we have just shown the top 5 or top 10 that are significantly differentially regulated.
Just to add to that, the one other difference, I'm not sure if that came across, is so the questions that we asked in our cohort is quite different from what Dr. Vijaya was talking about. What they did was they followed up children to see who goes on to develop active tuberculosis from latent infection. So they were not using it. So they were basically looking at correlates of risk. And what are the biomarkers that identify people, children who are exposed to TB, but who not develop TB, who then go on to develop TB. What we are doing is, children have already developed TB. Now, can we confirm that with just, not using clinical radiological diagnosis alone, but with biomarker diagnosis. So the children are not exactly equivalent. So I think there will be multiple differences there.
So can
I add one thing on the EDO? So we know we have studied the various pulmonary infection models, and there we have seen that EDO expression is a correlative of the vectoral persistency. So from the work of this pediatric tuberculosis presentation, I can only infer that EDO can be used as a correlative of the latent infection or persistent infection. That is my stake. Yeah.
I just wanted to add one thing. Did you look at the nutritional status of your children? Because that's going to make a lot of difference. I mean, we miss tuberculosis in the OPD and clinics.
Yeah.
Because
So
of the.
absolutely, I think it's a great question because the issue with, again, as I mentioned with host biomarkers is that they are always influenced by your co-infections and comorbidities. And obviously undernutrition, malnutrition is a huge problem. And there will be factors that are influenced by that. So what we had to do was, for the most part, we matched children based on age, gender, as well as with variety of parameters looking at stunting and wasting and so on, so that we had equivalent in all the groups. And the second thing we did was we did a multivariate regression analysis to control for the other factors as far as we could. But again, we cannot exclude every single thing, but nutrition was not a factor that impacted these differences that at least it was.
Maybe let's go back to the audience and then I come back to you, Amit.
Two types of tuberculosis in pediatric tuberculosis. So do you think there is possibility that the AFB negative or culture negative tuberculosis in children, it could be associated with some other pathogen which is still not identified and you could not detect by the methods that we use here? Is there possibility of some other unidentified pathogen to be there producing similar symptoms as TB?
Yeah, so that was the whole point, right? So you're asking if these children are not confirmed to be TB microbiologically.
Yeah, so do you suspect the possibility that there could be some other pathogen?
So that gets included in our unlikely TB group, because what happens is a lot of these children, you get typically put on empirical antibiotics for bacterial infection, for instance, or it could be a self-limiting viral infection. So these children are followed up to see whether the symptoms continue. So if it's TB, the symptoms will not go away. So they'll continue to have cough, they'll continue to have wasting and so on. So that is basically how the clinical diagnosis is done. And that is how you club the non-TB, non-confirmed group as well.
Okay, thank you.
Anybody else from the audience? Yeah.
Anil sir, and the question is, like you have said, that one second. Like oral drug delivery system is far much better than the taking 3 ml injection every day. So what if we have an implantable drug delivery system which maintains sustained release of drug for a period of like 1 week or 2 weeks, and we have to take only half of the dose which we are taking during oral drug delivery system. So what is your view on this? Like I have read a paper which is of Avinash sir from RCB and Vinay sir from CCMB, and they have described the hydrogel method using as an implantable drug delivery system for tuberculosis.
Oh, that's a very nice question. Basically, instead of having once an oral daily pill or injectables once every day, how do you see giving an implant to a patient which takes care of, they don't need any drug every day monitoring. That's how right now, if you look at HIV field has evolved from multiple pills, 10 to 20 pills in certain cases of HIV in extreme severe resistant phenotypes of HIV in 1990s, early 1990s, to 3 or 4 pills lifelong. And then it became in early 2000 and late 2000, early decade of 2010 around, it became what we call single pill. Now that was a fixed dose combination. Now that pill has basically gone into what we call long-acting injectables. So you give an HIV patient or even schizophrenic patients, you can, because they have the compliance problem, you can give one single injection as a depot, and that lasts for 2 months and in some cases 6 months. So this is feasible for TB, that you simply give one shot and leave it for 6 months. But that takes, it will evolve after 20, 30 years. By the time you have developed new drugs, I think sure you will be the one who discovers this long-acting forms of TB drugs. So it is possible, it's definitely possible. Sorry for the long answer.
For Sarnam, sir. So like you have showed the whole journey of detection, like first gene expert MTB, rifampicin assay, and if it is positive, then we will go for MTB DR plus assay for antibiotic resistance checking, and then for MTB MJIT for 960 cultures. So like as per my knowledge, it will take a few weeks to be to say that it is positive. So what we needed at this point of time to detect MTB in very early stages, like which technique or which type of technologies we needed for early detection?
Gene Expert gives you result in 90 minutes. So that is the fastest. There is another technology which has been approved kind of, it is very soon it is will be rolled out, which probably can detect in 1 hour. So that is again molecular test of course. The phenotypic test, there is no phenotypic test so far which can detect earlier than MJIT 960. That is the earliest, which usually takes about 2 weeks, 11 to 15 days.
Also amplified the dead bacilli DNA.
True.
So can we have something like extracting mRNA or something from the TB, and then
lot of work is going on, and very soon some technology they will be developed, which I am saying about the 60-minute test, which is probably maybe tomorrow, maybe declared by the health minister, or maybe in few months or so. This will be based on mRNA.
Okay, I'm being asked by the organizers to close the session. So thank you very much to all the speakers. It was very enjoyable, and all the audience. I think I'll hand over to the anchor. Yeah.
I thank all the speakers and the session chairs for this very exciting and informative session. I now request the session chairs to felicitate the speakers. Dr. Anil Kaul to felicitate Dr. Sarman Singh. Dr. Anil Kaul to felicitate Dr. Subhash Babu. Dr. Anil Kaul to felicitate Dr. Vijaya Lakshmi Valori. Thank you, sir. Dr. Shinjini Bhatnagar to felicitate Dr. Amit Misra and to Dr. Tamsenidis. Thank you, ma'am. I now request Dr. Amit Awasthi to felicitate the session chairs, Dr. Anil Kaul and Dr. Shinjini Bhatnagar. With this, thank you, sir. With this, we end our first day of the symposium. I thank again all the keynote and session speakers for the insightful talks on TB and detailing on how clinical research is very important in reaching the end TB goal. I thank all the participants for their wonderful participation. The breakfast buffet for tomorrow will start at 7:30 a.m. and the day 2 of the symposium will begin with a keynote address by Professor Ardre Stein at 9 a.m. The group photo of all the participants will be at 2 p.m. just before lunch to the left of the auditorium. I request all of you to kindly proceed for dinner arranged near the dorm area located to your right. I request everyone to sign the banner as a token of pledge to end TB. I request the concerned transport volunteers and committee to coordinate the transport back to the respective hotels. Good night.